Good morning, people at home. Looks like the sound is working this time. I'm just going to take attendance real quick and then we'll get started. Don't forget that if you are in the cohort that should be here and you're tuning in from home, someone just needs to call in. Good morning. Okay. Let's get started. I'm going to do a quick summary of what we're going to talk about today. We're going to keep going with the respiratory system. That's yesterday. Day 12. Okay, so right at the very beginning here, I'm actually going to start off. Did you guys watch the ATP video yesterday? Hopefully you did. The one where it talks about sort of like the origin of like our energy system, taking an organized sugar, breaking it down, building an ATP. Yes? Okay. So really cool video. It is applicable to almost all living things on Earth. So almost every single living thing uses that ATP system for energy. We're actually going to start off by watching this video, which is a really cool demonstration of what happens when you don't get enough oxygen. So what happens when you desaturate your red blood cells? There isn't enough pressure of oxygen in order to actually transport oxygen to your cells. And the really interesting thing here is that I want you to try and connect this to the video that you watched yesterday. So keep in mind that ATP reserve, that reserve of adenosine triphosphate that is all the different cell functions are using for their task, is a very small reserve. It's only 5 seconds worth of reserve. So if you stop sending oxygen to cells, within 5 seconds you use up that ATP reserve inside once you break the chain, and then nothing functions in your cells any longer. They actually completely cease functioning at that point, which is really kind of cool. It does not take long for cells to like completely lose their function. Now some cells are more sensitive to this than others. Your muscle cells can be anoxic, like with very low oxygen delivery for an extended period. You could literally take a belt and cut off the blood supply to your leg for 4 or 5 hours. No problem. I mean, I shouldn't say no problem. It'll be extremely uncomfortable. And when you release the belt, there's all kinds of metabolites and stuff that would build up in your leg, and cytokines and things like cell communication molecules. And they cause a great deal of pain and stuff once you reperfuse your leg, and it can cause blood clots and all other kinds of things. But that being said, the tissue does not die. It does not become necrotic after 4 or 5 hours of being anoxic, which is really cool. Muscles are very, very flexible when it comes to oxygen, mostly because they have an extremely high oxygen consumption rate. So when you're exercising really hard, if you can't deliver enough oxygen to your muscles have to still do something. And so they have separate anoxic pathways that don't use oxygen in order to continue to produce energy. So you guys have probably actually experienced this before. When you work out, when you're running or something beyond your oxidative capacity, so at a harder intensity than you can deliver oxygen to your muscles, what happens? What happens when you sprint eventually? I mean, can you just sprint infinitely? Just like run into the distance? Like what happens? You get out of breath. So that's your body saying, I cannot deliver enough oxygen to these muscles. That's exactly what it is. That's out of breath. It's saying, it basically forces your body to discontinue work so that you can provide additional oxygen and rebuild up that ATP storage. But what happens to your body? What's happening at the muscular level when you're sprinting? Can you just keep going? Just keep running forever? What happens to your muscles? What happens? They get sore. They cramp up, right? Like you cannot produce power forever from your muscles, right? And that's partially because of the buildup of the byproducts of that anoxic, that oxygen-free metabolism that they do. It's pyruvate getting turned into lactic acid, which you guys talk more about this in grade 12 if you get into the biochemistry of this. It's not really important for this course. But you start building lactic acid instead of the normal products that you get from regular oxidative metabolism. You guys have heard of lactic acid before, right? Lactic acid buildup in your muscles. So that's what's happening when your muscles are working without oxygen. They start to build up lactic acid, and they can still produce energy for a while. You can sprint at a lot higher capacity than you can actually deliver oxygen to your muscles for a while. But then that pathway breaks down eventually. You can't do it infinitely. That's fine for muscle. Skin cells, super resistant, no problem. A lot of your internal organs, pretty cool without oxygen for a while. But you also have a number of tissues in your body that are extremely sensitive to low oxygen, that can't survive for an extended period without oxygen. And the most sensitive ones, and the ones that are reflected here in this video, are your neurons. So your nerve cells are very, very oxygen dependent. And if you are without oxygen, going to your brain, for example, for 5-10 seconds, well, we already know what happens. What happens when you completely cut off oxygen supply to your brain for a couple seconds? You guys know this. What happens? You faint. You go unconscious, right? Even if you stood up too quickly and you weren't able to perfuse blood to your brain, even for a couple seconds, 2-3 seconds when you stood up really quickly, immediately your neurons cease to function properly. They can no longer coordinate movement. They can no longer coordinate thought, and then you become unconscious. So neurons are very, very sensitive to low oxygen. 2 minutes without oxygen, they start to die. So it's a lot different than muscle tissue, which is highly resistant. Even heart muscle tissue, which doesn't have a great anoxic system, like you're right, it's similar muscle tissue. It's not exactly the same as your skeletal muscles, but even that is fairly resistant to low oxygen conditions. If you're having a heart attack and you're blocking off oxygen supply to your heart, it keeps beating for a while. You don't just like instantly die when you have a heart attack, when you block the blood vessels to your heart. It'll keep beating for minutes afterwards. Not necessarily very well, so it doesn't necessarily function excellently, and it does start to die fairly quickly because it has a very high oxygen requirement. But it's fairly resistant, but not brain tissue. So we're gonna, like I said, we're gonna watch this video in a second. We'll get to it in a second here, And I'm just gonna like pause for the people here so that people at home can watch. Just give me 1 second, and I'll let you know when. Just so you can see what this is like. Okay, it's really interesting. Anyway, we're gonna watch that, and then we're gonna talk. We're gonna do this note together. Where is that? We're going to discuss a little bit more about the respiratory system, what it's for, how it works. Okay, we'll talk about the anatomy, how you breathe. And I think that is everything for block 1. Is that right? Let's see here. Oh, and then the last component of this, I have a video. It doesn't let you watch it embedded for some reason, but this is a bronchoscopy procedure. So this gentleman is having a camera and a little probe placed into his respiratory system to take some biopsy samples. I'm not sure why, he doesn't actually explain in the video, but it's great because you could see all of the different anatomy of this. What do the vocal cords look like? What do they look like when you're talking? What do the actual bronchioles and bronchi look like? Like it goes right in there so you can see it. Now this video is a little bit wandering. So if you don't care about like the anesthetic and you don't care about like, you know, the other parts of this procedure that are not necessarily the anatomy part, you can just skip over those. Okay, so don't feel like you need to watch this from beginning to end. Definitely skip around. But there is a section in there that is a great view, just following it in. And even the nasal cavity, like you never get to see this. When you get to like look up somebody's nose and go all the way around down to their respiratory system, I think it's pretty cool. So you can actually see what it looks like inside there. Anyway, so that's there for you. In learning block 2, did I get everything there? Yeah. In learning block 2, we're going to spend some time discussing what happens when this system breaks down. So what are some respiratory system disorders, some ways that your bronchioles or your alveoli or whatever can malfunction and cause you problems, okay? For some reason, I left off this component from the digestive system. It doesn't really matter, but we're gonna talk about some illnesses of the respiratory and the circulatory system. This links into the assignment from the end of the unit, where you guys are going to choose a technology for either the diagnosis or treatment of a condition or conditions of the respiratory, circulatory, or circulatory, digestive system. Okay, you can pick any disorder you'd like. You can pick any technology you like that's either for diagnosis or treatment of. Okay, we're going to talk about that project at the beginning of learning block 2. Some people had mentioned to me that they want to get started early, and that's fine. That kind of makes sense. You guys are coming up to the holiday, maybe you don't want to do it. Maybe you don't want to have it accidentally overlap with your holiday or your working your stuff. I personally don't want you to work on this during the holiday if you can get it done first. So we'll chat about it at the beginning of learning block 2. We'll go through that just so you know what to expect at the end of the unit. Anyway, so we're going to do that. That's pretty much all we're going to do in learning block 2. And then I attached this little video. I talk a little bit about asbestosis in the video. What am I talking about? In the lesson. And so if you're interested, this is a cool video that's like a video advertising asbestos from the 1950s. It's like miracle material. It's really interesting. Just so you can see what they actually used asbestos for. Everything. It was in everything, including the school. Man, the school was packed full of asbestos. We kindly had it removed 2-3 years ago. So it was actually in the school up until very recently. The only asbestos we've got left is actually the floors. So, but all the other asbestos is gone from the ceiling and the insulation and stuff. The floor asbestos is not particulate asbestos like they use for insulation. So this is not a respiratory risk, which is why it's still here. So we're not that concerned about the floors, but all of the respiratory problematic asbestos has been removed. I think eventually they'll probably get rid of the floors too, but it's extremely expensive, so it wasn't a priority. Anyway, I've got that. And then, have you guys ever watched, I can't remember, I'm sure I haven't given you any of these yet, these Chubby Emu videos. Have you seen any of these? So what these are medical case studies. It's a great channel. My wife put me onto this. She's a family physician. And it's basically just, here is a story about something crazy that somebody did. Well, they're not all crazy. Some of them are accidents. And then following along how that person's illness was diagnosed and treated. And for the most part, he does a good job of putting things into lay terms, like not using like highly technical medical language. Now, there's a little bit of that. But for the most part, he breaks stuff down. Now, I will say that if there are some moments in all of these videos where it kind of devolves into like a little bit of techno jargon, you could just ignore that part. It's not the majority of the video. But it does connect a lot of the things that we're going to talk about. So I try and find these for each of the systems. I had a digestive system one, but I guess I forgot to put it up. Anyway, this is about somebody who tried to eat some laundry pods for the views or whatever, and accidentally respired some of it. So got that laundry pod into their respiratory system. What's the effect of that? How did they diagnose and treat that? It's actually very interesting. So I'll try and post more of these. They're really cool videos if people are interested. Anyway, that's at the end there. Okay, so as I mentioned, I'm going to play this video here in the classroom for you guys. I'll do it on the Chromebook. And for the people at home, this is a good time to watch this because I'm going to refer to it in the lesson. So I'm going to just put this on pause for the people at home, and then I'm going to put this up. All right, we're back. Hopefully you had a chance to finish the video. Thoughts, questions, comments before we move on?
If you were to go right to 60, would you die right away? Or would it slowly lose oxygen more and more and then...
When he says you would have died, he says very quickly you would have died. What they mean is that essentially you're helpless, right? You've become unconscious, you have no way to correct it. And so yes, it's not like you just instantly all of your brain cells are going to start dying. But it's going to continuously desaturate until you die. So it does lead to death, yeah. but it's not instant. It's not instant. So it is progressive.
If you were to go to the extent of the cells almost dying but not dying yet, would that strengthen the cells?
Interesting. Would they become resistant to oxidative stress? Is what you're asking. That is a really interesting question. There's no mechanism that I'm aware of. There are cellular mechanisms that lead to stress resistance. For example, heat stress. So if you continuously expose tissues to increased temperatures, they do build up heat shock protein is what they build. They build up proteins that enable them to deal with heat stress better. But anoxic stress, I'm not sure. Definitely for muscle. So your muscle tissue does all kinds of things when you regularly give them anoxic stress. Not like this. You're doing it through exercise, right? So when you exercise and you're basically giving low oxygen to your muscles through intense exercise, you get many adaptations. You build more red blood cells, so your blood becomes more rich in red blood cells. That's probably the first adaptation you get. You increase the amount of blood volume. You increase the amount of blood vessels that are actually running through your muscle tissue. The way that your blood vessels are wrapped around the muscle fibers increases. There's more wrapping. That's called tortuosity. You increase the muscle tissue in your heart and the ejection fraction of your heart, which is how much blood you can actually pump per stroke out of your heart. So there are all kinds of adaptations that your body makes to anoxic stress from exercise. I don't know about if you get those adaptations from exposing yourself to low oxygen this way, but for example, if you go and sleep in a hyperbaric chamber, okay? So you intentionally go in a chamber at night that reduces the pressure, not this much, but reduces the pressure around you a little bit so there's less oxygen in the air around you, it will actually cause your blood to produce more red blood cells. It's the same thing as if you were going to go live at Machu Picchu or in the Himalayas for a while. It's assuming you don't go up too high, which could potentially be fatal. But if you go up a little bit, you actually build more red blood cells. So your body adapts to the altitude over time. So yes, for muscle. I don't know about neurons. I would assume at least, actually I can't say that. I don't know. Great question. Let me look into that one. That's a really good question. I have to look into that. Does anybody have any other questions? It's an interesting video. No? Okay. So let's talk about this system in a little bit more detail. Oh boy, this thing's chugging. I'm gonna have to reset this. There we go, much better. Okay. Respiration. All plants and animals require it. At least they require oxygen for respiration. Many protists require it. Some bacteria require it. So if you remember way back to the very first unit, we talked about how some bacteria are aerobes. They're obligate aerobes. In other words, they appreciate an oxygen-rich environment. That's a lot of bacteria and archaea that appreciate that. Some are anoxic. So some either can't deal with oxygen or... So it's not universal that oxygen is the thing that's used as a substrate to produce energy, okay? So oxygen and sugars. So in animals and plants, universally, we use oxygen for that. But there are some species, especially on the bacterial end of things, that don't require oxygen or actually are basically poisoned by oxygen. They use some other substrate for their metabolism. Now they all produce ATP. All of them are using that substrate to produce ATP, but not all of them require oxygen. But anyway, all plants and animals need that. So I can say that without reservation. So you guys know in basic how this works. It's a chemical reaction, right? You're using oxygen and sugar or fat, and you're producing ATP, which is the energy molecule, carbon dioxide, and water. Okay, that's cellular respiration, which I have the formula for right down here. Equation for cellular respiration. You guys have seen this before. I'm assuming this is nothing new for you, right? Previous to this, we've discussed, I think, in the first week, the idea that where is this happening in your cells? The cellular respiration in each cell. Where do your cells actually do that chemical reaction? You guys remember? Where does all the power come from? The power is the ATP. The powerhouse. No? Really? Yes. Mitochondria. Okay, it's the powerhouse of the cell, right? It's doing this chemical reaction. That's the purpose of your mitochondria. Okay, so they're doing this respiratory chemical reaction. I have glucose here as the other substrate, so we have O2 and glucose. Keep in mind that any hydrocarbon really can be used by the body for this purpose. So it could be fats, right? You could be oxidizing fats. It doesn't have to be sugars. They can be inserted into the same chemical pathway and used for energy. You guys know that already. You can use fat for energy. It's also possible to take proteins and with a little bit of jigging, get them into a state where you can also use them for energy. That's a state, a metabolic state called ketosis, where you're using proteins as energy molecules. In general, that is a starvation state, at least for animals. It is a starvation state. So we generally are only using ketosis when the body does not have access to fats or carbohydrates for energy. It's basically like an emergency energy reserve. And usually in order to get that energy reserve for ketosis, you have to start pillaging some of the protein structures in the body. In other words, you have to start disassembling things made out of protein to use them for energy. That causes muscle wasting if you do it for a long time. In other words, your muscles shrink. So, but in general, this is going to be fats and sugars that are used for that process. And it's all just to produce this magical molecule right here. So this is an adenosine triphosphate molecule, an ATP. And it's made up of an adenine. The actual precise structure of this you don't need to know, by the way. That's a grade 12 bio that you get more into detail on the structure of this. But in general, the components of it I'm going to talk about in this course. So you've got adenine. Now, you guys have seen adenine before. What's adenine from? Hopefully you remember. You guys have seen this. Where else do you find adenine? And maybe thymine and cytosine and guanine. In DNA, right? It's a sugar. It's a ribose sugar, or deoxyribose sugar, which is used to make. It's one of the components of DNA. These molecules are preserved, right? They're used for multiple functions inside of the cell. So adenine is also used as part of ATP. Adenine's actually pretty important. And then it's got a ribose attached to it, which is a sugar. So you would have seen ribose as well. Where's the ribose? It comes in two forms, ribose and deoxyribose. I'm not trying to trick you. Do you remember Fiagra? It's also in DNA, right? Now this is ribose, not deoxyribose. Deoxyribose is what's in DNA. Ribose is what's in RNA. So that's the difference between RNA and DNA, is one has deoxyribose and one has ribose. They basically look exactly the same, just one has a different, one of those groups around the outside of the ring, one of them is slightly different in deoxyribose. It's missing an oxygen. That's the only difference. So we got an adenine and a ribose, and then you guys have seen phosphate in grade 10 chemistry, right? PO4? Yeah? Sounds familiar? It's a polyatomic ion. So if you have 3 of them attached, that's how you actually store energy in ATP. And all that happens is all the different little proteins and stuff in your cell, if they need energy, what they do is they slice off, that's this right here, it's a little slice, the 3rd phosphate. When you cut off the 3rd phosphate, you release energy from the ATP molecule. And whatever is bonding to the ATP molecule can use that functional energy when it breaks the phosphate group off. So for example, every time you contract your muscles, the actual fibers, the protein fibers inside your muscle cells are grabbing ATP, breaking off one of the phosphates, and then that is causing the protein to physically move. It changes conformation, and that's what causes your muscle to contract and get shorter. It's a really interesting process. So it's actually breaking this molecule apart in order to function. That's how the energy exchange works. So this little molecule, the reserve of this molecule is what is keeping you alive. Okay, every single one of your cells has a little reserve of this ATP inside. And as long as you keep giving it oxygen, then you maintain that reserve, and then your cell stays alive. What's happening in the mitochondria is when you're doing this process right here, as you go through it, as you do this chemical reaction, the energy that's released from this chemical reaction is being used to build this molecule, to add a phosphate group back onto it again. Okay, so it's grabbing this same molecule, but with only 2 phosphates on it. That's called ADP. It is adenosine diphosphate, di for 2. And it's adding the 3rd phosphate group onto it. Okay, so it just keeps cycling between being a diphosphate and a triphosphate. And in that way, it's holding on to this energy reserve in your cells. So I like to think of it sort of like a water wheel. Okay, this is my analogy for this. Okay, so what you're doing is you're doing cellular respiration, right? And that cellular respiration, that's this right here, is pumping ADP, which is the low energy molecule, and it's pumping it up to a higher energy state. It's like filling a tank at the top. Okay, that's cellular respiration. You're filling your ATP store at the top. Then you're allowing the ATP to flow back down to ADP. Okay, that's the lower energy state. And as you do that, that turns a water wheel, and that water wheel is all of your different cellular processes. Okay, so and it's a continuous cycle. So you use cellular respiration to pump it back up to ATP, then it's converted back to ADP, allows you to do all of your cell functions, and then gets pumped back up by cellular respiration. So you can see how important cellular respiration is in this chain. If you stop doing cellular respiration, you stop filling the tank at the top, the tank very quickly runs out, 2, 3, 5 seconds, and all of a sudden you have only ADP in your cell and none of your cellular functions work any longer. It's really strange how it's such a small reserve. It really is extremely small. Now you do slightly build up that reserve with training. So especially, like I said, if you're incurring a lot of oxidative stress on your muscle, especially in this particular case, if you're doing high intensity training. So sprinting or if you're doing lifting, for example, if you're doing a lot of heavyweight low reps is the kind of work that will improve your ATP storage. So the size of this pump, how big the pump is in your body, is what's known as your VO2 max. That's how your capacity to refill your ATP storage. And a whole bunch of different things contribute to your VO2 max. VO2 max just means volume of O2, volume of oxygen, max, your maximum volume of oxygen that you can transport in your body. Okay, and I mentioned some of those things before that improve with training. How much your blood you can pump, how many red blood cells you have in your blood, your total blood volume. how wrapped your blood vessels are around your muscle tissue, all of those things. So all of those things contribute to your VO2 max, and so it goes up considerably when you train. When you're born, you generally have quite a high VO2 max. Babies are very physically fit, which is interesting when they come out. So your VO2 max is actually quite high. And if you monitor VO2 max in the population, a really interesting trend that you notice is that as people age, their VO2 max tends to go down and down and down and down and down over time. And that is not necessarily a natural state for the human body. It has to do with the fact that people don't exercise as they age, and so then they tend to lose their oxidative capacity as they age. This is much more common in Western culture than it is necessarily. It's not like a default for humans or anything, but in Western culture, we have a really high tendency to very quickly devolve our VO2 max over our lifetime. People tend not to exercise here. So if you look at, for example, an elite athlete, okay? Let's say that you're like an elite cyclist. You can measure somebody's oxidative capacity. You may have actually seen this on the internet or on a TV show where people are like on a bike and they're like wearing those masks, or they're on a treadmill and they're wearing like a mask that's measuring the gas that's coming out. So you can actually measure how much oxygen they're using in each breath. And so I wish we had the equipment here. It'd be so cool if we could do that test here. Anyway, if you're an elite athlete, usually your VO2 max is somewhere around 80 mils per kilogram per minute, but that's very high. Okay, that takes a lot of training to get there. That's elite level. There are ways to approximate this too, by the way, by looking at running times and things like that, but they're not exact. So what that means is that you are using 80 milliliters of oxygen, or you can use 80 milliliters of oxygen per kilogram of your body weight. Okay, so this is by body weight. Size matters in terms of your body size here. Per minute of time, okay? So 80 mils of oxygen per minute per kilogram of you. A sedentary person, that's a person who essentially never exercises as an adult. And by the way, keep in mind, as a baby, it usually starts off considerably higher. It's usually somewhere in the 50s as a baby. But a typical sedentary adult usually has a VO2 capacity of about 35 milliliters per kilogram per minute. That's not a good VO2 max, by the way. But that's a sedentary adult. And it is extremely typical in our population to have that as a VO2 max. If you're that level of fitness, you're not necessarily going to be winded from climbing a flight of stairs, but you're probably going to be winded from climbing 3 flights of stairs, which probably applies to many people that I know. So, like I said, that's probably average for the population as an adult. If you're ill, okay, you actually have respiratory problems, and this is something like you actually are winded from climbing a flight of stairs. Maybe extremely winded. Maybe you can like barely make it up a flight of stairs. That's usually somewhere below 20. So if you have less than 20 mils per kilogram per minute, you're starting to get into some serious trouble. Okay, so a number of different problems can cause a really low VO2 like this. Asthma, well, actually not asthma, but COPD, if you have chronic obstructive pulmonary illness, you're having difficulty actually pulling the air into your respiratory system. It could potentially reduce your VO2 max severely. Usually your ability to breathe does not restrict your VO2 max. Usually you can breathe, like actually get the air in and out, pretty much as you need to exercise. That's not what's holding you back. It's not how much you can breathe. It's usually something on the other end, which is like your ability to pump blood or how much oxygen your blood can hold or whatever. That's usually the limiting factor, not how much you can breathe, except if you have something that is actually physically limiting your ability to breathe, like COPD, or if you have, which we're going to talk about later, emphysema or something like that, an actual scarring of your alveoli or something like that, in which case, yeah, that is going to start affecting your VO2 max at that point. Anyway, I think all this stuff is interesting here. The reason I'm discussing this stuff is because this is why you're breathing, okay? This is the purpose. The reason that you need to get oxygen into your body is to produce ATP and keep you alive, right? And you got to transport it to all of your cells. So I want to talk a little bit about how the oxygen actually gets in. How does the oxygen go from out here in the world into your blood? And it's a really simple process. It's just diffusion. So you guys have talked about diffusion. I can't remember if we had talked about it in the course in the first week. This is the problem with me being gone for a week. Did we talk about diffusion at all at the beginning? I can't remember. No? Okay, I'm going to do a quick little demonstration here. I've got a diffusion demonstration. Where is my physics thing? Here we go. I think once you see this, I think you'll be like, yeah, that's obvious. That's obvious that that happens. Okay, so let me put a bunch of particles on this side of the barrier. Okay, this is just a simulation of particles of gas. What's happening in this room right now, and by the way, this is true for any fluid, which means including liquids. Liquids and gases both exhibit these properties. Okay, but if I were to remove the barrier here, what would you predict would happen to the particles that are separated between the two sides? I think this is probably not something that is like super archaic here in terms of understanding. What do you think would happen? Right, they're going to evenly distribute. Does that make sense? Right, I remove the barrier and we can do that. So if you actually look at the number on each side, oh, can I show that? Where's the number? Center of mass. Where is it showing that? Oh yeah, the center of mass is across the bottom. That's how it's distributed between the left and the right. So you'll notice very quickly, this just comes to an even distribution. And this is a physics simulation. So keep in mind, it's not intentionally placing more on the other side. All that it's doing is just looking at the collisions of the particles and making them go wherever they would go when they bump into each other. That's it. Okay? So it's mimicking what real particles do. It's not forcing them to one side in this simulation. It's just simulating the actual forces on particles. So over time, you just get an even distribution. This is the principle of diffusion. Okay, that's all that it is that when you have a higher concentration on one side, it's going to evenly distribute. There's a force that is causing particles to want to evenly distribute across a certain space. Okay, this is like the same thing as spraying perfume in one corner of a room. If you do that, those particles are going to want to evenly distribute in the room, and so that the smell diffuses into the room. Okay, the particles move around within the room. This is the reason, by the way, that you can live in this enclosed room. Because think about it. You're sitting there and you're expelling carbon dioxide into the air right in front of you, right? But you're not building up a pile of carbon dioxide right here in front of your body, okay? It's not sitting there. When you breathe the carbon dioxide out, it is diffusing out into the room, and the room is ventilated. And so the carbon dioxide is continuously being removed from the room. But if this didn't happen, you'd literally build up a little block of CO2 in front of you, which you'd continuously inhale, right? But that doesn't happen. Question?
Does the mask like kind of hold any CO2?
Does the mask? Great question. No, it doesn't. And these, I should actually put these studies up. There have been a number of studies looking at whether there is a rebreathing of CO2 that happens with masks and other health effects of masks, and there really are none. It is problematic if the mask that you're wearing is impermeable, as in you're basically wearing like scuba gear. And in that case, you do need to make sure that it's properly being ventilated. But with a mask like this, you actually get diffusion of the particles right through the mask, and there is no accumulation of CO2. So that's a really good question, though, because people do talk about that. There have been a number of studies done on that, and so there is not significant CO2 rebreathing occurring with the mask. Good to know. So I think what's less obvious is let's reestablish the divider, okay? But let's have an even number of particles on both sides, okay? This is less obvious, but what do you think would happen in this case? Now it's not so obvious. It's not a vacuum on one side, right? Where there's no pressure on the other side. Here the pressure is even on both sides. What do you think will happen if I remove the barrier now? What do you think? Okay, good idea. Okay, let me remove the divider. Now, it's not as rapid, okay, when you have even pressure on both sides. But what you will notice, and this is purely based on the physics of the particles, is that over time, and you can see the center of mass for each tank here, is slowly shifting towards the center. If I let this go, eventually, why don't we we can watch it, it won't take us a couple seconds. And again, keep in mind that this is just a physics simulation. It's not forcing the particles to go in any particular direction. They're just bouncing off each other. But you notice over time that it does equalize. It does equalize over time. And that's just due to random collisions between the particles. Okay, that's the principle of diffusion. That in a fluid, You always get movement from high concentration to low concentration until the concentrations are equal, okay? So this is really key to an understanding of how your lungs work, how you're actually getting oxygen into your body. So let's talk about that. We cool with this? And notice that once it gets to parity here, it basically maintains it, right? I mean, it's not perfect. It's random collisions, right? There's no force holding them in any particular direction, but it more or less stays even on both sides, which is just due to random chance, which is really cool. All right, so let's talk about the implications of this. So again, diffusion here, a good definition to know here. Gases and liquids, so this is all fluids, always travel from high to low concentration, always. This principle, the principle of diffusion, is probably responsible for, I would say, the majority of biological processes in your body. So great corollary here. We talked about the digestive system, right? You've got high sugar concentration and fat in your small intestine, right? You've eaten all the food, you've broken it down, it's in your small intestine. I think I mentioned this in the lesson, but why do you think that those nutrients are going into your blood through the villi and the microvilli on the small intestine? Why do you think they actually go in? Hint, hint, it's related to this. No one wants to take a shot at that, eh? Okay, well, I'll draw it for you. Imagine you've got your villi, or microvilli, and the microvilli has got this little tiny blood vessel running into it. And then you've got some sugar molecules here. Oh, that's a horrible looking sugar molecule. Let's try that again. By the way, these are absolutely not to scale. Okay, so like let's say that this is our situation where you've got some inside of the blood vessel and lots outside the blood vessel. Where is the force pushing these things? This is just simply principle of diffusion. Which way are they going to want to flow here in this case? Which way are they going to want to go? Right, they want to go from high to low, right? Into the blood. This is the reason why these nutrients are making their way into your blood. They're not pumped in. There's nothing on the surface of the cell that's grabbing sugars and forcing them into your blood. That does not exist. So the only reason that this works is simply by diffusion. So in order to get this to work, you can see what would happen very quickly. You would end up getting a balance, right? So how do you keep sugar going into the blood then in this case? How do I keep pumping it in? I can't physically pump it. I can't grab the sugar and force it in. That mechanism does not exist. How would I keep this going? Keeping sugar flowing into the blood. What's something that I could do? Okay, I'll give you a hint. Is the blood inside of this blood vessel stationary? Is it just sitting there in a pool? What is it doing? What does the blood do? Exactly, it's flowing out, right? It's flowing. So as this blood is flowing out, it's carrying these, whoop, I lost some of my sugar molecule there. It's flowing, okay, well now this is gonna become problematic, but it's flowing away, okay? And it's carrying those sugar molecules away and replacing with low concentration sugar in the blood, which maintains this gradient between the two of them. The key thing here is the gradient. So the gradient is the net direction of movement. You want to keep that pressure up that's pushing in one direction. So now if I remove those sugars, then now there's going to be less sugar on this side, and then the gradient's maintained and more sugar will flow in. You remove the sugar, you maintain the gradient, and more flows in. Okay? So in the digestive system, this is the reason why sugar and fats and amino acids are flowing into your blood. As long as you keep pumping them away and using them and storing them somewhere in your body, there's going to be that force that's constantly pulling them in to your circulatory system. Okay, that's just diffusion. The exact same thing is happening in your respiratory system in order to keep everything going. Okay? So what happens is you need to maintain a high concentration of O2 in your alveoli. do I need to go over the anatomy of this again? It was in the video, but maybe I do. Do you guys remember what alveoli are? Okay, maybe I'll take a quick side trip here and go through the anatomy really quickly again. But those are the little gas exchange sacs. I'll give you a chance to write this down. Did you already write this down? You guys are fast. Cool. Okay, let me jump over to the anatomy part. Let's talk about this really quickly here. So you probably remember from last year, air is going to enter either through the mouth or through the nasal cavity. Yeah, passes by the oropharynx. The oropharynx is just the back of your throat, and then the pharynx is just below that. There's a little flap here, the epiglottis. I should use another color on top here. A little flap here, the epiglottis, that can potentially block the upper part of your airway so that you don't swallow food into it. Or I should say block the lower part of your airway. And then you've got the trachea right here, right below your larynx. Then there's a split. These are your two bronchi. And then the bronchi are going to split into smaller and smaller passageways called bronchioles. Those are the little pathways here. And then eventually, once you get right to the end of these, you're going to start seeing these little bulbs. At the end, I think I've got a blow up of it right here. At the end of a bronchiole, you're going to come to a chamber with these little spherical buds on them, and those are the alveoli. So the alveoli are extremely thin, only one cell thick, and they are thin because they are allowing exchange of gases to go across the membrane. When I say allowing exchange, that's exactly what they're doing. They are just simply, it's basically just like a screen that is allowing gas to pass across the membrane. There's no pumping happening here. So they're just simply allowing the gas to go where it wants to go based on the principle of diffusion. So going back to the previous page here, let's take a look at what this actually looks like on the level of the alveoli. So inside the cell, we've got high oxygen concentration. Those square brackets, by the way, around the O2, do you guys use these in chemistry yet? I don't know where you are in chemistry, if you're even taking chemistry. Square brackets just means the concentration of, okay? So when you put that around any chemical, you're just referring to the concentration of that chemical. So the concentration of O2 is high inside the alveoli. How do you keep it high? Because it is flowing. The O2 is flowing into the blood because there's not very much O2 in this blood that's returning to the lungs. So the diffusion is forcing it to go into these little tiny capillaries that are going around the alveoli. How do you keep the oxygen concentration high in the lung? What do you have to do? You got to breathe, right? That's why you have to breathe. So you are replenishing the O2 concentration inside your alveoli. That's why you're breathing, okay? So as soon as you stop breathing, you stop replenishing the O2 concentration inside of your alveoli. And once you do that, this is instead of being high O2, relative high O2, this will just become the same concentration of O2 as in the blood, right? These are going to equalize over time due to diffusion. What happens when they equalize? What happens to the movement of oxygen once you have the same amount on both sides? You guys just saw this. If I had like a 15% concentration of O2 here and a 15% concentration on the other side, what happens to the movement across the membrane when it's the same? It slows down or it stops, right? You get no transportation. So this entire system of diffusion completely breaks down as soon as you stop ventilating, replacing the oxygen at the alveoli. That's why you got to breathe. Okay, so breathing is pretty important in this process. Now keep in mind that if you were to stop breathing now and just hold your breath, it's not instantly going to equalize, right? It takes some time for you to get an equalization across that alveolar membrane. And so you're not instantly going to stop transporting oxygen and become unconscious. You guys know that. It doesn't happen the moment you stop breathing. But over time, you do get less and less and less concentration inside the alveoli until they equalize, at which point you will become unconscious. There's pretty much no way around it. The other thing that's happening simultaneously here is The same thing, but with CO2. So keep in mind that the blood that's coming to the lungs is full of CO2 from your cells. Your cells are producing that as the waste product from respiration, right? So you've got high CO2 inside of your blood vessels, and you've got low CO2 in your alveoli. And so again, naturally, you're going to get this movement of CO2 into the alveoli, and you have to keep breathing in order to remove the CO2 in your alveoli. If you stop removing the CO2 in your alveoli, then this concentration inside the alveoli becomes high. If it's high on both sides, you stop removing CO2 from your blood, at which point that's going to cause big problems too, because CO2, when it's in your blood, would you guys have talked about this in any other course? Probably not. When you dissolve, maybe in grade 10, I doubt it. Do you guys know what happens when you dissolve CO2 into water? How it's stored in water? This might help. You guys have drank this before. I'm sure you've had a carbonated beverage, something that has CO2 dissolved into the water. What does it do when you carbonate it? Oh, maybe you don't know this. You might not know what this is. You'll see it as an ingredient on the actual soda can. It says the ingredient on it. Oh, maybe not. So it's on the soda can, it says carbonic acid. Carbonic acid is one of the ingredients. And the ingredient, the reason that there's carbonic acid in there is that's just dissolved CO2. So when CO2 is dissolved into your blood, it creates carbonic acid in your blood. Now, a little bit's fine. That's normal. You're supposed to be transporting CO2 in your blood. But the more CO2 that builds up in your blood, the lower the pH. Remember, lower pH means more acidic. So your pH becomes more and more and more acidic over time. And there's one particular cell in your body that really does not like low pH, and that's your neurons. Again, your neurons, sensitive to everything, such a pain. But they don't like low pH. So the moment you start getting really high CO2, you start getting low pH, and then your brain cells stop functioning properly. Interestingly enough, your brain cells also hate high pH. So if you do the opposite of this, let's talk about that situation. What would happen if I hyperventilated? Okay, you guys have probably done this before. Oh, okay, better not do it too much. Okay, you guys have hyperventilated before. What does it feel like when you hyperventilate? What do you feel like? You get lightheaded. What's actually happening is your neurons in your brain are not functioning properly. That's what's happening. You're actually impairing the function of your brain cells. And the reason that you're doing that is because you're actually removing too much CO2. So if you do the opposite, if you make the concentration of CO2 in your alveoli really, really low, in other words, you get rid of all the CO2 by hyperventilating, what happens is this gradient becomes super, super big. Okay, all of a sudden you have all of this force pushing CO2 out of your blood, and you remove all of the CO2 from your blood, which causes your pH to go up in your blood, the opposite. Okay, you're losing the carbonic acid in your blood, and your pH changes dramatically. And then that causes you to become dizzy. Your brain cells do not like extreme high pH either. Now, when we're talking about extremes, we're only talking about like 0.5 on the pH scale. Okay, your body only has a very small range where it's tolerable. If it gets way, way too high, you become unconscious. Like if your pH changes by 1, remember that's times 10 difference in pH. That you will become unconscious. If your pH goes down by 1, you will also become unconscious. So there's a pretty small range in the middle that your body likes. Conversely, and this is really interesting because we talked about the pulse oximeter, right? And about being 100% saturated. Right now, assuming that your respiratory system is working correctly, your blood is 99 or 100% saturated with oxygen. In other words, you are carrying the most amount of oxygen in your blood as you can possibly carry. You are 100% saturated with oxygen. What do you think happens to your oxygen concentration in your blood if you increase the O2 concentration on this side, inside the alveoli? It's interesting, thought-provoking question. You're already 100% saturated. You have the maximum amount that your blood can carry already. You're currently carrying 100%. What do you think happens if you increase the pressure of oxygen going into your blood? What will happen then? So that's an interesting idea. Would it come out of solution in your blood? That's an interesting idea That is not, in fact, what actually happens, but that is a really interesting idea. The reason that that doesn't happen is the amount of energy that it would take for the oxygen to come out of solution is too high. In other words, you'd essentially be going above like the vaporization energy for oxygen for it to come out as bubbles. And there's not enough energy in this system in order for that to actually happen. So you can't force that. You can't force the air to come out of the oxygen to come out of solution unless you very, very rapidly decrease the external pressure. And then you can cause oxygen and nitrogen in your blood to come out of solution if you very dramatically decrease the outside pressure very quickly. That's called the bends. You guys have heard of the bends before when you go scuba diving and you like come up too quickly? So you can do that, but not under natural conditions. You'd have to change the air pressure rapidly in order for that to happen. But you can't saturate it anymore. So what happens in reality is nothing. Okay? So you can't get more O2 into your blood than 100%. It's maxed. So you guys may be putting a connection here together, which is that there's no way to get too much oxygen into your blood. There's no way for your body to know if you're breathing too much, right? Not through oxygen, right? Because if you're breathing too much, let's say you just go outside, just start breathing a lot, you know? There's really no way for your body to know based on oxygen if you're breathing too much. How does your body know if you're breathing too much? The oxygen system doesn't change.
Based on CO2.
It's all based on CO2. Everything is based on CO2, which is really interesting. Your entire regulation of your respiration is completely based on CO2. Your body very carefully monitors the CO2 in your blood, but it does not monitor the oxygen at all, not even a little bit. And so that's why as your oxygen concentration goes lower in your blood, like Destin is a great example here, he has no mechanism of feeling that. There is nothing in his body saying, you are in an emergency situation. You don't have enough oxygen. I mean, he's experiencing symptoms like tunnel vision and stuff like that, but he's not feeling an urge to breathe, okay? It's not, his body is not saying, breathe more, breathe more, breathe more in this particular case, because the breathe more feeling has nothing to do with oxygen. You can't detect how much oxygen is in your blood, not naturally, okay? But you can through CO2. So when the CO2 builds up in your blood, that is the signal that is telling you to breathe more. When you go for a run outside, you're not detecting the oxygen concentration going down in your blood. And in fact, the oxygen concentration does not go down in your blood. You're able to keep it at a reasonable level always in your blood while you're exercising. What you're detecting is high CO2. That's what's telling you breathe faster, breathe faster, breathe faster. It's all based on CO2. So this has an interesting side effect. The fact that you can't detect oxygen, but you can only detect CO2. And that happens if you were to ever hyperventilate, which really, really decreases your CO2, and then hold your breath. So what happens when you hyperventilate and hold your breath is that you've removed all the CO2, right? Pretty much all the CO2 from your lungs. You made it zero. And so very slowly, the CO2 is going to start building up in your lungs. But the oxygen in your lungs is going to dissipate fairly quickly, right? You're holding your breath, you're not bringing in new oxygen, but you cannot detect that, okay? So because you hyperventilated and the pH is taking a long time to equalize due to the CO2 concentration, you don't get the urge to breathe, and then you become unconscious. So hyperventilating followed by a breath hold is a terrible idea. Don't do that. Those two things don't go well in combination with each other. And it is a mega no-no when you are snorkeling. It's okay to take a few breaths before you snorkel dive, but you don't want to hyperventilate. You don't want to hyperventilate for a minute and then take a dive as a snorkeler. Because what happens is you'll be swimming around underneath the water. You won't feel like you need to breathe because your CO2 level is still low. And you're unconscious. You just suddenly will become unconscious under the water, which is a terrible place to become unconscious. So just as a heads up, that's all because you cannot detect the oxygen level in your blood. If you ever, you can see these videos on YouTube, but you can watch people do training for snorkeling and for deep diving, and they hyperventilate in order to reduce the urge to breathe. And you will have to have spotters. So people are always spotting all along everywhere in the pool for people becoming unconscious while they're doing that, because obviously it's a terrible place to become unconscious, especially if you're alone. It's an easy way to die. So interesting. Did you guys already write this down? I can't remember. You look at this? So this is all based on diffusion. I'll give you a second here. High concentration in the alveoli of oxygen, low in the blood, means oxygen is continuously diffusing into your blood. And the exact opposite is true for CO2. High CO2 in the blood, low in the alveoli, means CO2 is continuously going to go out. As soon as you stop breathing, you stop doing that. Or at least you progressively stop doing it. So, leading to unconsciousness. I mean, pretty much the big takeaway from this lesson is don't stop breathing. And then you'll be good. If you're really into kin, you're really into study of movement and stuff like that, human movement. I was really big into kin in university. A lot of it is focused on gas transport for obvious reasons here, because gas transport is like sort of the basis for allowing all of that movement to occur, at least on a biochemical level. So if you're into fitness or whatever, it's all about oxygen transport pretty much. You guys okay? You good? You can never tell. Some people type really fast. All right. So I just want to really quickly mention here, this is not necessarily on the respiratory end, this is on the other end, but the opposite is true at your cells, right? So you need to transport this gas through your blood to your cells. The exact opposite is happening at the cellular level. So as long as you have high O2 in your blood, right? This is at the cell level. You put it into the blood. So as long as you have high O2 in the blood, it's going to diffuse into your cells where there's low O2. You're using up the O2 in your cell, so it's low in there. So you always have oxygen going into the cell in order to keep it alive. And that's, again, just purely by diffusion. So no pumping is occurring here. This is just natural flow of gas. And same thing, as long as you continue are ventilating, you're going to have low CO2 in your blood, right? This is after the lungs. After the lungs, there'll be low CO2. You removed it. And so the CO2 is going to naturally diffuse out of the cells and into your blood, which is what you want. You want it to remove the waste product so it doesn't build up inside of your cells. So the exact opposite process is happening at your cells. Then you send the blood back to the lungs, the gas exchange happens, and then you send it around again. It's a continuous cycle. It's a loop, if you haven't figured it out yet. The blood is going in a continuous loop through the lungs. Lungs, tissues of the body, lungs, tissues of the body. Heart in the middle. That's true for mammals, by the way, and a lot of other animals. There are some simpler animals that don't have that good of a loop. We've got a really cool closed circulatory system where it's literally pipes, blood vessels transporting things around. But if you're a grasshopper or something like that, you basically just have like a big chamber full of fluid. They don't have blood vessels per se. And instead of having a heart that pumps it through tubes, the heart is more like just like a fan, basically, that's kind of waving the fluid around on the inside and keeping it kind of going around the inside of it. It's not, there's no tubes inside, which is kind of interesting. So they have a kind of a simpler system. Any questions about this before we move on? Okay, so that's the basic principle here. That's why you're remaining alive in this room. Okay, I want to talk really quickly here about the structural components here. So structure and function, that is sort of the main thrust of this entire unit. What are the structures and what do they do? And the four main structures that all respiratory systems have to include are these four, okay? So we focused in on this first one already, this idea of you need a thin membrane for gas exchange. That must exist somewhere in the system. Grasshoppers have that too. They don't have lungs, but they do actually have holes in their exoskeleton that allow gas exchange with the fluid underneath. So it's not the same as a lung, but they actually breathe through the surface of their body. But you got to have a thin membrane somewhere to allow gas exchange to occur. You can't support a multicellular organism. In general, you have to have a large surface area for that gas exchange to occur as well. So it's got to be thin to allow the gas to go across, and it has to be of a large surface area because you have to transport a lot of gas. So we do that in our bodies by having spherical alveoli. You guys saw the structure. A sphere has the largest surface area to volume ratio, and we want lots of surface area. So it's a perfect shape for them to evolve into because they have the most surface area per unit volume. So the maximum amount of transport across the membrane. You find the same structures on the surface of gills. So there are spherical structures on the surface that also allow gas exchange to go between water and blood in that case instead of air and blood. The same thing is also true, by the way, at the cellular level. So your capillaries, which are the little tiny blood vessels that are at the cellular level, they are extremely small and they are cylindrical. So they also have a very high surface area on the outside to allow for high gas exchange. So very small and cylindrical also has a very high surface area to volume ratio. You need to actually be able to move the respiratory fluid, the gas transport fluid, around. Okay, so I'm gonna state that as high blood supply. Okay, it also means in terms of high blood supply that you need to have lots of vessels to potentially carry this fluid around. For you have so many cells, trillions. So if you've got this many cells, you actually have to have blood vessels going near every single cell. There's actually no cell in your body that's more than 4-ish cells away from a blood vessel. So the blood vessels go everywhere. They're going in between all over the place. They're in every single tissue, all permeating everywhere. Okay, usually no more than 4 cells away. The only exception there is you have this interesting transition area at the skin where the very surface of your skin, the keratinized cells, the dead skin cells that are at the very surface, there's sort of like an intermediate layer that's just below that is sometimes more than 4 cells away from a blood vessel because those cells are becoming dead cells that are becoming the surface of your skin. So there's like a transition area, but in most places in your body, they're no more than 4 cells away from blood supply. And you got to have some system for ventilation. You got to move the gas around across that membrane surface. Even if you're a grasshopper, oftentimes they will move their carapace around in order to change the gas, exchange the gas that is on the surface of their body. So that there's some system in place that's actually causing the gas to be ventilated around the exchange surfaces. If you're a fish, well, you guys have seen it before, they move their gills around, kind of flow water over top of them in order to increase the ventilation. That's ventilation for them. And you guys have to breathe. So those are some key components that you always have to have in a respiratory system for any animal. Yes?
When you yawn, is that because like you have low oxygen?
Great question. I've heard that as well. And I've never investigated it. I have no idea. That'd be something interesting to look up. I'm not sure what the function of that is. I wonder if that's true. Maybe. Any questions about this? No. Let me know. Okay. Really quickly, discussion of what are the parts in there and what are some main structural features of each of them. I'll do them one at a time here. So first of all, and you may have talked about some of this stuff in grade 10, your nasal passage, that's the thing at the top, right? Nasal and oral mucosa, this is in your mouth as well. Most people are not mouth breathers, where they're just breathing with their mouth open all day, but you can do it. You can breathe through your mouth all day if you want, especially if there are various things that can be obstructive in your nasal cavity that prevent you from breathing well. But in general, people are breathing through their nose. And in that nasal passage, you may have noticed it's wet. Okay, it has mucus in there. The purpose of the mucus is to capture and filter fine particles. So there is a reason why it's wet inside your nose, and that is when it's sticky and particles will land on there and they get filtered out so that you don't breathe those particles in. You don't want them inside, especially the particles like virus particles and other things that potentially could make you ill. You've also probably got some hairs in there. Some people more than others, but the hairs are a filtering mechanism. They filter out large particulate matter from respiration. You don't want those in your lungs. You've also got an epiglottis. I think this is probably mentioned in the digestive system video. In other words, a physical barrier that prevents swallowed food or liquid from entering your respiratory system. So it's a physical barrier. Don't want food in your larynx. It's probably happened to you before. Had something go down the wrong pipe. It's kind of uncomfortable. Question?
If the epiglottis is like stopping stuff like that, I didn't watch the video yet, but how did like the laundry pod get to that place?
So that's an interesting situation. I'm sure you've choked on a little water before. So part of what happens when you severely irritate something in your respiratory system, even through vapor, or even severely irritate something in your digestive system in the tube, it causes you to aspirate a little bit of the liquid, like you kind of go like that, and a little bit can potentially slip by into your respiratory system. For the laundry pods specifically, it only takes microliters, a very, very small quantity, because it's extremely caustic. When you watch the video, it's disturbing how dangerous it actually is. But yeah, it doesn't require much. So even if you just aspirate a tiny little amount, it can do an insane amount of damage. So you are aspirating small amounts of liquid into your respiratory system all the time, right? Little bits, little tiny bits. I mean, how many times have you done that? That's fine normally when that water is of relatively neutral pH or whatever. But if it's highly caustic and you're aspirating a little bit, then it's a completely different story. Yes.
So like what happens when the water goes like down and then right goes and stuff? Like how does it?
Most of the time you can purge it.
Just like back out?
Yep. You've done it many times. You've had probably a respiratory infection where you've had lots of mucus and liquid in your respiratory system, and you get it out, cough it out. Most of the time. Obviously, if a lot is entering, then it becomes more problematic. But yeah, you do have mechanisms. For the liquid, you're mostly coughing it up. For the solid particles, well, we're going to talk about that in a second, but you have some other systems that are in place to remove solid particles. Large pieces you have to cough up. So if it's a really big piece, like the size of a marble or something, that you got to cough up. Smaller particles, though, there's another system, which we're going to talk about right here. So if you go down into the trachea, so now we're going below the larynx, you have a couple of structures in there. One, you have these cartilaginous rings. You can feel them if you run your finger up and down your trachea. And those rings hold open your trachea all the time. So that it's a solid physical structure that keeps the tube open. Your esophagus doesn't have that. So when you're not swallowing food, your esophagus actually just collapses. It doesn't stay open. But your trachea has these rings, and the rings always hold it open to ensure that you can get gas down there. If you've ever been punched in the throat and had a temporary collapsed airway, or maybe a more severe collapsed airway, it's very distressing. This tube needs to remain open and very open in order for you to breathe properly. And the other really interesting structural characteristic of this tube, of the trachea, is it has something called a ciliated epithelium. So if you look at it, the cells actually have these teeny tiny little hairs on them. They're called cilia. And they can grip small particulate matter and physically push the matter, almost like a millipede, like a millipede's legs, pushing the matter back up the trachea until they go slightly above the glottis or the epiglottis, and then you can swallow them into your digestive system. So they actually pass moderately large particles of food, and they just kind of walk the particles back up, and they allow you to remove particulate matter from your respiratory system. This is especially true for fine particulate matter, so things that are like 10 microns in size. Those are things that are basically invisible. Dust. Like you can't see all the dust in the room. There's a ton in the room right now. You can't really see it, but you're constantly removing this stuff with your ciliated epithelium out through the trachea. And this ciliated epithelium extends all the way down right to the end of the bronchioles. It doesn't quite go into the alveoli. The alveoli can't have this on the surface because it obstructs gas exchange. So once you go right down to the alveoli, there's no ciliated epithelium, but it goes pretty deep. And you can use that for removing particulate matter. The really, really small particles, things that are like 1 or 2 microns, are actually quite dangerous because you're not very good at removing those, and those have a tendency to accumulate and cause scarring in your respiratory system. So very, very fine particulate matter. Sometimes that can even be absorbed into your blood if the particulate matter is that fine, 1 micron. That's a very, very small piece of particulate matter. So we try our best environmentally to restrict the release of very, very fine particulate matter because it can be quite problematic. Anyway, so like I said, the rings hold open the trachea, and the ciliated epithelium grabs and removes particulate matter. Interesting side note here, when you smoke, or if you smoke, hopefully not when you smoke, but if you smoke, you paralyze your ciliated epithelium. The gases in cigarette smoke actually paralyze your ciliated epithelium and make it so that it cannot remove particulate matter from your lungs, which is one of the reasons why smoking dramatically increases your chances of getting a respiratory infection, because it's actually paralyzing your system that you're using to remove particulate matter and bacteria from your lungs. Just as a side note. It's not permanent. It paralyzes it for a couple hours after exposure to cigarette smoke, but that's certainly enough to have negative health consequences for people. And then the last physical feature I want to mention here. Oh, sorry, I skipped over one. Your bronchi and your bronchioles also have this ciliated epithelium. Oh, and mucous membranes. I missed the mucous membrane part. Sorry. The mucous membrane part is that they are producing a sticky fluid that is also there. It's like flypaper. It's to trap particles. So the ciliated epithelium can transport the mucus back up. It has no problem moving the mucus, but it's sticky enough that it grabs little particulate matter and stuff that makes it into your respiratory system. In general, assuming that there's not an insane amount of particulate matter in the air that you're breathing, this is a very good system. I mean, it can consider this, like, assuming you're not exposed to something, significant during your lifetime, you're filtering out this junk that you're breathing in all the time. If you've ever woken up in your bedroom, there's like a little light streaming in, and you can like see, particles like just kind of like flying around in the air in your room, and that's very common. You're breathing that in all the time, pretty much your entire life, and your lungs are continuously removing it, and usually it has no effect on you. So, which is pretty amazing. Like, you're removing this particulate matter constantly. And then the last aspect here that I wanted to mention is this idea that your alveoli are one cell thick. So they're very thin, they're spherical, and they have a ton of tiny, tiny blood vessels running on the outsides of them. That is, they're highly vascularized. Vascularized just means has high blood supply. And all of that is in order to maximize the gas exchange between what's inside the alveoli and what's inside the blood. So I would say in general, these are the really important structural features that you should know about your respiratory system. Sorry I'm talking so much. I feel like I'm talking a mile a minute today. Apologies. It can be a bit much sometimes. You guys good? Okay. Yeah. Okay, cool. Let me know if I'm going too fast. I have a real tendency to do that. Okay, last thing I want to mention here before we go, before we take a break. This is my last thing, right? Yeah. I just want to talk a little bit about the mechanics of breathing. How you actually get the air ventilated into your lungs. So, first of all, before we actually go in and discuss it here, take a breath. Think about what you're physically doing with your body in order to get the air into your body. First of all, when you breathe in, what are you doing? Think about it for a second. Yeah.
Your lungs are like expanding.
Okay, so your lungs are expanding. What's causing them to expand? You're right. Absolutely, your lungs are expanding.
So, like more air going in.
There is more air going. What's forcing the air to go in? What's making it expand? This is not immediately obvious. I don't think it's obvious either. What do you think?
Is it the diaphragm that, like, pulls it in?
Can you feel it when you breathe in? It's almost unconscious.
Yeah.
But you are. You're pulling down on your diaphragm. Okay? So it is an active process. Breathing in is an active process. You have to do something to expand your lungs. Now when you exhale, just have a feel when you're exhaling. What are you doing to exhale? It's hard because you never think about it. You never even experience the feeling of it because no one ever thinks about it. But what are you doing in actuality to get the air out? Anyone want to take a shot at it? Before. So in this case, it is related to the diaphragm. The pushing down part is the breathing in. It's hard to feel, but when you constrict it, you're actually pulling the diaphragm down. Your lungs expand.
Into the diaphragm.
the diaphragm gets a little bit smaller. It's a muscle. You're contracting it, and the diaphragm's getting smaller. Your diaphragm is attached to the bottom of the chamber here, right? So when you contract it, when you're breathing in, what you're actually doing is you're pulling the diaphragm down. You're flattening it out.
Is it the ribs that expand then?
So it can be related to the muscles in the ribs, but that only happens when you forced expel. So when you have air in your lungs and you go, if you do it, you'll feel, you have to contract something in here. Those are the muscles in between your ribs. They're called intercostals. So when you're forced exhaling, you need to contract them. But right now, try it. Try breathing in just a little bit. And then breathing out. What do you have to do to breathe out? You're not contracting a muscle in your ribs. In fact, you don't need to contract this. What do you do?
You relax your diaphragm?
You relax. You're doing the opposite, okay? So exhalation is a passive process at rest. When you're exercising, it's not passive any longer because you need to ventilate more. And you actually have to force air out. And you're right, you need to use your intercostals at that point to force air out of your respiratory system to keep ventilating. But at rest, you don't need that. At rest, all you have to do is relax. Your lungs are actually elastic. So if you inflate the balloon and then you just stop inflating the balloon just pushes the air out. They're elastic. So the first thing to mention here is that inhalation is an active process. To bring air into the system, you have to expend energy. You have to contract a muscle to get air in. This muscle, by the way, is what becomes paralyzed in an opiate overdose. And the reason I'm mentioning this is because we're having a huge problem with this right now. You guys may have heard of people like ODing on, what's the big one that's happening in the news a lot? Fentanyl. You guys heard of this? So what's really happening here is it's causing paralysis of your diaphragm. You're no longer able to breathe in because the opioid is causing diaphragm paralysis. You're losing your ability to inhale. And what's happening in that case, so you contract your diaphragm, this muscle that's attached to the bottom of your thoracic cavity, and it pulls down on your thoracic cavity and creates a low pressure inside your thoracic cavity. Okay? So you've got a low pressure inside your thoracic cavity and consequently inside of your lungs, because your lungs are adhered to the inside by negative pressure to the inside of the cavity. They're not stuck to it, but they are, because there's essentially a vacuum in there. Your lungs are stuck up to the side of the chamber. And if you have low pressure on the outside and you have high pressure on the inside, diffusion, we just talked about this, which way is the gas going to flow if you have high pressure on the outside and low pressure on the inside? To the low pressure, right? That's the principle of diffusion. This is diffusion as well. So once you create that state with low pressure on the inside of your lungs and high pressure on the outside of your lungs, gas will naturally flow. Which is what this says right here. I'll let you fill that in. And I have a little demonstration of this. Okay, so some balloons inside a sealed chamber here, okay? And there's a hole in the top. This is your trachea. And we've got a flexible membrane at the bottom to create negative pressure, okay? So if I pull down on this membrane, you can see that it's creating negative pressure inside here, which high pressure on the outside, low pressure on the inside, it's going to cause gas to flow in. Okay, and then the same is true if you relax. I mean, these have already deflated because they're elastic, right? Just like your lungs are. But you can also force exhale if you push on the chamber and create high pressure on the inside, okay, by either relaxing the diaphragm or contracting your intercostal muscles, essentially pushing your ribs in. Okay, you're going to create high pressure on the inside, Now there'd be low pressure on the outside, lower, okay? And then you're gonna get the opposite. Oh, this one's leaking a little bit, which is kind of, there we go, that's a little bit better. Can you guys see that? One thing to mention here is just like your thoracic cavity, there is a space in between your chest and your lungs. It's a very small space. And it's sealed. It's sealed up in this chamber. So as long as this chamber is sealed in between the two things, if you pull down on here, it's going to create negative pressure inside the lungs because there's not a vacuum, but there's essentially balanced pressure in here that's causing these to be associated with each other, right? But if you ever put a hole in your thoracic cavity, like you are stabbed or you're in a car accident and something perforates your thoracic cavity, so we create a separation, okay, so now air can just go into your thoracic cavity. What ends up happening is this, which is now that you've separated the two, the outside of the chamber from your lungs, now when you create negative pressure in the cavity, the air is actually going to flow into the cavity and not in through the mouth into the lungs. Okay, and that causes your lungs to collapse. So that's a potential consequence of perforating your thoracic cavity. People don't talk about that, but when you get stabbed and it goes through, it's not only if it like perforates your heart, right? Obviously that would be the worst case scenario. But if you perforate the thoracic cavity, you do have to worry about this. So it's very difficult to breathe. There's a special kind of bandage. Anybody taking first aid in here? Yeah? Did you guys learn how to do the valve bandage if you have a thoracic perforation?
I don't think so, no.
Fair enough. Sometimes it's in first aid courses and sometimes it isn't. But there's a bandage that you can use that will allow air to come out of the thoracic cavity but not back in again. It's like a little valve that you can do. Okay, so this is the opposite condition here. If you relax your diaphragm, it allows air to flow out of the system. High pressure on the inside, low pressure on the outside. The elastic properties of the lungs do most of this. Keep in mind that your lungs have no muscle tissue in them. So your lungs themselves are not doing the ventilation. They're essentially, just like this, a balloon sitting in your thoracic cavity. They're not participating physically in the ventilation component. And I mentioned just at the bottom here, yes, you do have intercostal muscles. Those are muscles in between your ribs. And you can force exhale by contracting your intercostal muscles. Generally not done at rest unless you have severe respiratory issues, in which case you're having difficulty removing air from your lungs due to emphysema or COPD or something like that. So you don't usually use those intercostals unless you're either exercising vigorously or you have some type of illness that's causing you to need them. Which is, as you can imagine, is very tiring over time. They're not meant to be activated all the time. Any questions about the ventilatory mechanism here? Like what's actually happening in ventilation? So there's two questions I want to look into. One, what does yawning do? And what was the other one? Somebody else asked a really good question this morning. Oh, do cells adapt neurons to low oxygen? That's another good question. I'll have to see if I can find the answer to that one too. And of course, there's an awesome academic article on that very topic. read about this in a minute. Oh. This is just the last component of this. I already mentioned this, but probably good to note this somewhere. With CO2, when you combine it with water, it dissolves into water and produces carbonic acid, which is H2CO3. Do you guys remember how to write acids from last year? Where the hydrogen comes in front of the negative ion? Does this sound familiar? Grade 10? Maybe some of you didn't cover this in grade 10, I don't know. But anyway, that's carbonic acid, H2CO3. And this is what is regulating how much you ventilate. It's purely based on how much carbonic acid is in your blood. So high blood pH means you don't have very much carbonic acid, which means you don't have much CO2. Causes your body to naturally respire, to ventilate at a slower rate. And this exact opposite is true if you have really low blood pH, that is high acid content in your blood. It means you have high CO2, which means it triggers you to increase your respiratory rate, all based on the pH of your blood. Meanwhile, you have no way to detect oxygen whatsoever. It's kind of a strange system. If you do get into watching these chubby emu videos, I certainly did, but you'll notice a lot of this physiology becomes very, very important in medicine. Somebody presents to the emergency room and they're hyperventilating. They're breathing very quickly. Immediately, you know that that person has low blood pH. That's the reason that your body does that. And so then you have to start diagnostically thinking, why would this person have low blood pH? What is happening to them right now that is causing the pH to be low in their blood? Is it something chemically imbalanced that's happening? Is it some type of poisoning that's occurring? Are they experiencing stress that's leading to a high oxygen requirement and therefore is causing their pH to be low in their blood? Okay, all of these things. But in understanding the underlying chemistry of what's going on here helps you diagnostically to figure out what is wrong with the person. You need to know this in order to figure out what's wrong. And again, this comes up in those chubby emu videos. They talk about this. They're like, okay, well, this person must have low blood pH. How do we figure out that? Like, how do you treat that? Et cetera, et cetera. Any questions from anything that we've discussed this morning? Okay, I'm gonna stop talking now. I'm gonna come back after the break, guys, around 11:15. You guys can take an early lunch if you want. We're done. That's the morning content right there. I think is there a video that goes along with this? Yeah, if you're interested, you can watch part of this bronchoscopy. This is just to actually look at the physical structures inside the lungs if you're interested to see what they look like. You don't have to do it, but they're kind of cool. And then we're gonna come back after the break. And we're going to talk about some illnesses of the respiratory system. That's how we're going to finish up today. I'm also going to very quickly talk about the project at the end of the unit. You don't have to start it today by any means, but if you're interested and you're like, oh, I've got some extra time, I don't want to work on this on the break, I don't want to have this accidentally overlap with my break, you just know what it is, okay? We'll talk about it in more detail. All right, cool. You guys can just chill at home. I'll see you guys at 11:15. Hey guys, at home, welcome back. Just let me know in the chat if you're here for the second half. Get attendance out of the way. All right, right on. Why isn't this working? Hotspot turn off? Yep, it did. There we go. Cool. Okay, so in block 2, what we're going to do is we're going to spend some time, like I mentioned, talking about some ways that the respiratory system can fail or work incorrectly, some illnesses around the respiratory system. The note is, it's part of the same note. It's at the end of the note package, the one that we were already working on. And then afterwards, if you're interested, this one's really cool. This is a medical case study about somebody who's aspirated some Tide laundry pods. I believe they're Tide laundry pods. And an interesting video about the miracle of asbestos. It's interesting to get a sort of a historical perspective on asbestos, since it seems so taboo now, so unusual that asbestos was used prolifically for everything. Like if you've ever seen The Wizard of Oz, do you guys see The Wizard of Oz? Yeah. The snow in The Wizard of Oz is asbestos. Like that's like a thing. They use it for everything back then. So anyway, really quickly, I'm actually going to go over the requirements for the project in case anybody's interested in getting started early. Keep in mind you do not have to start it now. So if you're busy or whatever, or for whatever reason, you don't need to get started on it yet, but you're welcome to. Some people were interested, so give me one sec. I'll pull this up for you. So the assignment for this unit is called the Medical Technology Presentation Assignment. What you're going to do is prepare a presentation. You're not going to give the presentation, at least not live, but you are going to record it. So you're going to do it. You can use Loom, or you can use some other like screen capturing software. If you don't want to use Loom, you're interested in some other screen cap software, no problem. If you're not sure what to use, so you just talk to me. I'm happy to find you some alternatives. Okay, they definitely exist. But essentially, you're going to create it in a Google Slides slideshow, and then you're going to give the presentation through a screen capture. The difference between this one and the other one is here there's going to be a focus on where is your information coming from. And you're going to be completing a research sheet, which you're also going to include as part of your assignment. Please don't forget to include the research sheet, okay? That's a big part of this, is you showing where you're actually getting your information from. So the research sheet should look like this. You don't have to use this one. I've linked this one. What? I need permission? Oh. Personal account, that's why. Let's try that again. There we go. So you don't have to use this research sheet. It just needs to contain this information. But the idea here is that you're going to give me the full APA citations citing for your source. Okay. If you're not sure how to do APA citation, you could try using the Cite This For Me Chrome extension. This is probably the easiest way to do it, where it's literally like a button in Chrome that you click and it will give you a full APA citation for whatever website or resource that you're using. If you're reading an academic article or whatever, it just cites it for you. It like checks through the article for all the information and just creates a citation. That's a shortcut. That's easy. Or you can look up literally any guide on APA citation. There is a website called Citation Machine. That also gives you the option to scan. You like put the URL into it and it'll scan through it and find all the information that you need for your citation and format it properly. Or it just has the instructions right on there explaining to you how to make an APA citation. What information do you include in what order? But just make sure that you're getting in the habit of doing this, okay? Don't just put a website down for your citation. You must include the full APA citation. Pretty much anywhere in academia, anywhere, college, university, anywhere you go, if you're citing an information source, you have to give more information than location. I found it here. You need to say when you cited it, what the name of the author was, etc., etc. More information is always required for a citation. APA is the most common in science. In science and any of like the technical industries, APA is favored. In the arts and in English, it's usually MLA citation. So I don't know if you've used that in English class before. You may have. MLA is really common in the arts, and APA is really common in the sciences. So I'm going to use APA because you'll see that in university. Almost all of them use APA. So you're going to do that, and then the key here is when you make this when you're researching. Okay, don't do this at the end. It completely defeats the purpose of it if you do it at the end. The purpose of this document is to separate your research from your final product. It's very easy if you're reading any source, and creating something written from that source. It's very easy to plagiarize. Very easy. You'll read it, and then when you write your version of it, it may be altered, it may have one or two different words, but you're essentially just copying and pasting something from a source. It's so easy to do. Even if you try not to do it, it's so easy to do. And so the purpose of this document is to separate your source from your product. And this is a really good habit to get into because anywhere you go in academia, you're going to have to very clearly delineate what is source and what is product. So here, I want you to try and write down only in point form, okay, the key information from your source. Don't have any sentences. You're looking for key points that you're gaining from your source. And that way, when you incorporate this into your final product, when you use this to create your final product, you are not directly copying and pasting ideas in full from your resource. Last quad, for some reason, I saw a lot of students quoting things. In science, you never quote. I cannot even think of in an academic source why you would ever quote something directly. It's very, very rare to quote in the sciences. In English, you quote all the time, right? In the arts, you quote. You're like, this person said this in this way, and the way they said it was important. That's something in the arts, but that doesn't exist in the sciences. The way they said it is not important. We're looking for facts here, factual information, and so that you're going to be putting into your own words. Okay, so keep that in mind. You're not quoting stuff. The other reason that you do this is for you to synthesize what you're reading. It's very easy to read some piece of information, and just write it down, word for word the same. Basically, your brain has to do nothing for that. It needs to hear it and then repeat it. It's like you're copying something directly. That is like the least engaging thing you can possibly do with your brain, and you're not really getting much out of the source material when you do that. There's going to come a time in your educational career, I'm assuming you're going to post-secondary at some point, which you may or may not. I don't care, whatever you want to do. But if you are going to have to read a source material, and you're going to have to actually understand the source material. You're going to have to take that and synthesize that information. And if you don't actively synthesize, if you don't write down what you're thinking while you're reading it, and put it into a context that you understand, it essentially just goes right through. You don't absorb anything from the source material. You're going to use that source material, then you're going to use another source material, and by the time you're like a couple source materials in, you've lost everything from the first source material that you read. You literally get nothing out of it. And so you have to make sure that you're using your time wisely in post-secondary. You'll never get the stuff done that you need to get done, and you have to synthesize things in order to do that. So here this is an opportunity to get a little bit of practice. When you're researching, think about what you're reading. What is the important thing from what I've just read, and how do I summarize that in a phrase or a sentence, okay? As opposed to just copying and pasting. So this is actually surprisingly important, this research aspect. It seems like it's just like a surface thing, but it's an essential skill in academia to be able to synthesize. So take some time and actually make this research sheet, okay? I realize it's not worth a huge amount in your final product, but it is valuable. Yes, question?
Do you have in-text citations?
What's that, sorry?
in-text citation?
Are you asking, do you include in-text?
Yeah.
No, you don't need in-text citations. Not for this, not for this. If I was gonna get you to write an academic paper, then you'd have to have in-text citation. For this, I'm just looking for, in general, what is the information you've gained and where have you gained it from? Yeah, I don't need to go in quite that much detail at this point. There's probably, I don't know if what we do in 4U Bio, do we do in-text citation? We used to have a project in 4U Bio where you wrote an academic paper about something, a review paper, and we used in-text citation for that. I don't know if we do that project anymore. I haven't taught 4U in a while, so, but not in this course anyway. You won't have to do that. Okay, so what are you actually doing? What is your presentation about? Over the course of the next couple days, we're going to spend some time talking about illnesses of the three systems. Okay, today we're going to do respiratory. I believe tomorrow we'll already be getting into the circulatory system illnesses. And so what you're going to do is, this project is an opportunity for you to demonstrate your knowledge of the three key systems. Okay, those are the three that we're going to discuss in class. And it's an opportunity for you to demonstrate the knowledge about a particular technology that you're going to research. You want to discuss the function of that technology, the history of that technology, and why it's important within this medical context that you're going to discuss. Okay, I'm going to tell you about what specific information I want you to include, but keep in mind that that's your goal here, is for you to explain your knowledge of the systems. So you need to show me that you're knowledgeable about the three systems, and you need to show me your knowledge of this particular technology. Okay, we're going to incorporate those things together. When you're creating this presentation, consider your audience, okay? Sorry, go ahead, yeah.
If we have to demonstrate knowledge of like the three systems.
Not just the three, sorry, you can pick a system.
Oh, okay.
Yeah, sorry, one of the three systems, one of the three systems. Good question. So you don't necessarily need to show your knowledge of all three systems, you need to pick a system. Now, you may pick a technology where there's an obvious illness in multiple systems, or there's a crossover between the two, and then that might be what you show, but it doesn't have to be all three. For level of, what level of explanation, assume that you're explaining this to a parent, okay, or somebody that hasn't taken this course but is in grade 10. I don't expect you to be using a level of scientific technology that is like far beyond your current educational level. But I mean, you should be using the scientific terminology that we're using in class, right? So attempt to speak to another grade 11 student and that you're describing this to, okay? A competent grade 11 student like you are. Okay, keep going through here. I already talked about the research document. Make sure that you're recording a video. You're not just submitting the presentation without the video component. I'm not marking the presentation. I'm marking your presentation, the explanation of these things. Okay, so you being in it is key. Not that it's just like a dead document. Okay, I'm trying to assess your understanding, and that's why you need to be the person explaining it. So you're going to have a title slide. Include those things. Tell me about the development of your technology. Okay, so and keep in mind that you're selecting a technology here that is either to diagnose an illness or to treat an illness, okay? It can be one or the other. Some of these things, I guess, could potentially be both, depending on what the technology is, but it only needs to be one. It's either a diagnostic or a treatment technology, and it must diagnose or treat at least one illness of the respiratory, circulatory, or digestive systems. Some of them, like if you're picking X-ray, could be applicable to all three. Maybe you'll talk about multiple illnesses, okay? But it only needs to cover one, specifically of those three systems. So don't pick something that is specific to the limbic system or something that's specific to the nervous system. It's got to be one of these three. Tell me about the technology. How was it developed over time? Okay, take some time with this. Okay, what was the idea here? Who invented it? Who was the person responsible? Why was it developed? So oftentimes these technologies are developed because of a specific need. What was the need that the person was attempting to satisfy with the development of the technology? Sometimes it's not its current function. X-ray wasn't necessarily designed for its current diagnostic purposes. What was it designed for? What's its purpose? And in this part, in the development part, you're going to give me a timeline of development. How has your technology changed and improved over time? The first X-ray device used for a medical purpose and modern X-ray technology is night and day different. those technologies are completely different. How did they change over time, and what were the specific changes that were added to it over time? Okay, there are quite a number of them. Okay, whatever you can find out about that. So you're trying to give me a timeline of development. How has this technology changed and improved over time? The second component is you're going to explain to me, how does your technology actually work? If you pick X-ray, for example, like, how is it taking a photo of the body? You know, what is that photo? How is it producing X-rays? Why is it using X-rays? Explain the technology, okay? Don't give a rudimentary explanation here. It's like, oh, it takes a photo of your body. You know, you need to go into way more detail here. How is it physically doing that? Okay, how is it generating X-rays? What do X-rays do? What's the purpose of using X-rays instead of ultraviolet or whatever other part of the spectrum? Okay, discuss how the technology actually physically works. Here in that part, it may be helpful, by the way, for you to look up a YouTube video on how it works. Most of these have very good YouTube videos explaining the function of the technology. MRI, X-ray, CT. All of those things have detailed videos so that you can find that explain very precisely how they work, okay? So make sure that you're explaining it. And then lastly, you need to discuss at least one illness in detail, okay? You can talk about multiple illnesses if you want, but you have to describe at least one illness of the three systems and explain the causes of that illness, the symptoms of that illness, and the treatment of that illness, okay? So those three things you have to discuss, plus you have to tell me, how is your device associated with the illness, okay? So for example, if it's a diagnostic piece of equipment, how is it used to specifically diagnose that illness? If you were to take an X-ray of that illness, of the lungs, what are you looking for? If you were to look at an X-ray of somebody with tuberculosis, let's say you're picking that as your illness, how do you know that person has tuberculosis from that X-ray? Like, what is the specific use of that technology for diagnostic purposes, okay? And that'll require a little bit of background. You got to figure out how this technology is actually used to figure out if somebody has that thing, okay? You might even want to ask somebody, ask a professional. You're welcome to do that. You can use a person as a source, okay? Anyway, like I said, there may be many. You may choose to describe more than one, but you want to go into great detail about one specific illness, okay? Take some time and really discuss your illness. Okay, we're going to talk about these illnesses over the next couple of days in a very cursory way, okay? Just the basics, but you're going to get into detail here in the last component of your project. Okay? So through that, it says, be sure to focus on an illness from these three systems. Use your knowledge of the anatomy and structures of those systems in your presentation, okay? Talk about specifically how your illness is specifically affecting the structures within that system. Okay, remember, you're trying to tell me about your knowledge of that system through discussing the illness. Okay, so keep that in mind when you're telling me about it. And then lastly, you need a works cited slide. That is secondary, keeping in mind that you also have the research sheet. So I have two places to look for your research information. Okay, when you submit, I need a link to your Loom presentation or whatever service you're using. Usually they provide a link. Sometimes it's an actual file, and that's fine. You can upload a file. I will say that the file uploading service with Brightspace is not fantastic. And on the download end, it greatly adds to how long it takes for me to mark them, because I usually have to wait 10 minutes for your video to download if we do it that way. It's really annoying. I don't know why that's so slow, but it's certainly not my internet at all. So, but anyway, it is problematic. A link is preferred, but you know, whatever you got to do, and that's fine. And don't forget to attach your research document. You have to include it. Any questions about how this works? I don't want to harp on this forever. And we're not necessarily starting this today, but you're welcome to if you'd like. Question?
When do you want like the due date to be then?
So I wrote the due date as Friday,
Yeah.
because that is a course day. I have to do that. And I realistically, I mean, you could finish it on Friday if you wanted to. I won't be marking these until at least Tuesday of the following week during the break. Okay, there's no way I'll get to them before then. So if you handed it in at Monday at midnight, I don't care. Go for it. I would not leave this for longer than that, because it's going to end up on my back burner after because I'm going to mark everything else, and then it's just going to end up getting marked way later, which you probably don't want. And you shouldn't leave it to the end of the break, because the moment you get back from the break, we're going to be starting on the next thing, and then it really will build. The end of the quad, it goes so fast after the break. So just as a heads up, don't leave this. Don't just let it sit out there. Okay, or at least I recommend that you don't. I don't want you to get stuck at the end. How long should the video be? Good question. There's no specific time included here because what I'm looking for is that you actually address all of these things. I've seen good presentations that were about 7 or 8 minutes long that included all of this information and were detailed enough. I've seen 5-minute presentations that didn't include the level of detail that they probably should have. Very few 5-minute presentations do. So this is somewhere around 8 minutes is probably what you're shooting for. So I've seen 10, 12-minute ones that were fine, that included all the information, maybe included a little bit of extra, and that's okay. If you're getting up above the 15-minute mark, chill, just chill, guys. That's probably too much information that you're including in your presentation. Also, if everybody hands in a 15-minute video, it's going to take me 12 hours to mark this, which I would really prefer that it did not happen. But somewhere in that range is totally fine. Okay, so the actual number is not super critical as long as you have the correct information. Somebody said, do we attach the research table to the slides? No, so yes and no. There is a works cited slide where you're going to include just your works cited. That's in APA citation. But there is also a separate doc that has each citation plus in point form the information that you gain from that citation. So it's really 2 components where you're including your sources. Any other questions?
Would you recommend us like doing like jot notes on each slide and then just like explain everything in further detail?
Your slides should be point form.
Okay.
Don't include all of your information on your slides like a text that you're reading or a script. If you want to include a script, like there's places for notes like underneath, that's fine. But I would highly recommend that you don't swamp your slides with text. The purpose of this, guys, is to prepare you for an actual academic presentation on a topic, which you do in university and grad school constantly, all the time. Okay, especially later when your classes get a little bit smaller. You don't do it a lot in the first years of undergrad because you have 500 people in your class. But later on when the classes get a little bit smaller, you do academic presentations like this constantly. And your slides, if they're super busy, if they're just covered in text, nobody reads them. Essentially, they're there for no reason. And they're not meant there to be there for you. They're there for the viewer to help them understand the thing that you're discussing. So even if you had a picture in place instead of text in some places, sometimes a picture is more appropriate. Here, look at this thing. I'm going to discuss this thing that you're looking at. That's more important than having text on a slide in some circumstances. So keep in mind that the slides are for your audience. They're not for you. Okay? You can have a script, but just it shouldn't be on screen. Loom lets you record for 5 minutes if you don't have the full version. But if you sign in with a student account, you get the full version and then it's unlimited length. So if you're having difficulty doing that, let me know. But recently I've looked into it and it still is free for students and teachers. So you should have unlimited video length if you're signing up with your school Gmail account. Any other questions? Okay. If anything else comes up, please let me know. Feel free to email. I'd be happy to discuss, but you don't need to start this today. All right, let's talk about some illnesses. For each of these, by the way, on the note, what you're trying to get out of each of these are the causes, symptoms, and treatments. You'll notice that this is the same thing that I'm asking you to discuss in your presentation, okay? Causes, symptom, and treatment. And it's not necessarily listed as causes, symptoms, and treatments on each of these slides. This is another synthesis activity. The idea is that you're listening for these 3 things, and that's what you're going to add in. If you're unsure at all, or I forget to mention one of these things, don't be afraid to ask, okay? I'll get it in there if I forgot. Present to Chromecast. Nope, I don't want that. What do I want? Present to external display. It sounds right. Yeah, there we go. Okay, so some basic information about respiration. We've kind of talked about this already. You breathe about 10 to 14 times a minute. You need to maintain that. That's at rest. And you need to maintain that in order to properly ventilate. So that means you breathe about 20,000 times a day. You inhale about 35 pounds of air, which is a lot of air. It's a gas, so it doesn't exactly weigh very much. And there's lots of stuff in it. We already mentioned this. So you have some mechanisms in place to help filter out a lot of those things, but I mean, they're not perfect. And so if you're overwhelming your system with a lot of particulate matter or whatever, it can lead to some of these illnesses. We're gonna talk about them now. So we've already discussed the anatomy, so I'm gonna kind of skip through this part here. Emphysema. So we mentioned that the alveoli at the very end of the chain, the last step, the site of gas exchange, is a 1 cell thick membrane that is a sphere. Well, if you have a 1 cell thick membrane, that is a very, very sensitive membrane, right? I mean, think of how thick your skin is, 50 cells thick or something like that. It's thick, all right? And then it's got that keratin, dead skin cell tissue on the outside, and then below that is like a submucosa and like a whole bunch of other layers. And this is thick. But your alveoli is super thin. It's only a few microns across, right? And so it's very, very sensitive to potential damage. Emphysema is when you damage it, okay? So for whatever reason, a number of different things can cause physical damage to the alveoli, but the most common is cigarette smoke. At least in humans, the most common is cigarette smoke. So prolonged exposure to the stuff in cigarette smoke causes you to damage your alveoli. Often your body will repair it. So it is possible to repair damaged alveoli, just like cells anywhere, they can reproduce. However, if you're consistently damaging it, which people tend to do with smoking, they tend to continuously do it for their entire life or for an extended period, eventually you get scarring. So scarring, once you get scar tissue across your alveoli, they're essentially gone forever. You can't repair that. So you get a permanent scarification of your, I've got some pictures later so you can see what it looks like, but it's a permanent scarification of your alveoli. And once you get scar tissue forming over top, they are non-functional. So first of all, they're not flexible, so you're not able to, they're not elastic tissue that you can use to properly ventilate. And then the second part of that is that they just don't allow gas exchange to occur. Even if you could ventilate them, no gas exchange occurs in the emphysemic parts. Okay, so that's called airway collapse. Airway collapse is when they essentially get sealed off from the rest of the lung because they're no longer functional as diffusing tissue, perfusing tissue. And you get little areas in the lung where air gets trapped, and you can imagine, yeah, you get stale air in there, right? They're not properly getting perfused with oxygen. You can get necrotic tissue forming in those areas where you just simply are getting no oxygen to the cells. There's still cells alive there, but they're not getting oxygen any longer. One of the symptoms of that is people tend to get a barrel chest. So they tend to hold a lot more volume in their thoracic cavity. You get barrel chest. And that's a compensatory mechanism. You're trying to increase the ventilation into your lungs in order to compensate for the fact that you have scarring in some of your alveolar tissue. And then so long term that happens. And then you get some other symptoms that would obviously be associated with the fact that you can't perfuse your lungs with oxygen. Coughing. Again, there's liquid that accumulates in the areas that are getting sealed off. You get shortness of breath. You can't exhale very well, because keep in mind that you've got essentially air pockets that are kind of locked away inside of your lungs, and the scarring is not flexible, so it's difficult to exhale. And the weakness, anxiety, confusion, all that comes from low blood oxygen. So you're just not able to get enough oxygen into your blood. You've removed the mechanism that you use to do that, and so your brain doesn't work properly. Your muscles become physically weak. You have low oxygen in your blood. The anxiety and confusion is because your neurons are not functioning correctly in your brain. And you can cause damage to your heart tissue, so it can cause heart failure. And then you get swelling around the lungs. So that causes edema. Edema is just fluid buildup in the bottom of the lungs. And then the end product of that is you could potentially get respiratory failure. Respiratory failure is when your lungs no longer perfuse enough oxygen to keep you alive. So eventually, if you damage them enough, you lose the ability to properly perfuse and keep yourself alive. So as I mentioned, smoking is the #1 cause here. There are pollution causes to this as well. They're not super common in North America, but there are places in the world with very high air pollution where emphysema is caused by air pollution. It's not super common in North America. It is in some industrial contexts here. It can also happen from exposure to hot air. So for example, if you were like a firefighter and you weren't wearing protective gear and you went into a room and the air is, 600 degrees or something like that in a room and you inhale that, it causes burns on the inside of your lungs and that can potentially lead to emphysema. Or potentially infection. So there are a number of different infections that can damage the alveoli and then cause scarring, which also causes emphysema. Not super common in North America. They do exist, but it's just not super common here. What can you do? Not much. So you can increase the total concentration of oxygen in your lungs by giving somebody supplemental oxygen. That increases the diffusion pressure to push oxygen across the membrane into your blood. That helps when you're not perfusing enough in. You're basically forcing extra oxygen across the membranes that are still functional. That's what the extra oxygen is doing. Now, if I put you on supplemental oxygen, the effect is extremely minor. Remember that you're 100% saturated, assuming you have a normal functioning respiratory system right now. But it could potentially be helpful for somebody with emphysema. Now, there does come a time when the scarification is so bad that supplemental oxygen doesn't allow you to perfuse enough to keep you alive any longer, and then it is fatal. That causes respiratory failure. And there really is no way to cure it. Once you have that scarification, it can't be undone. It is permanent. So this is a CT scan, or at least it might be one aspect of a CT, or it could be an X-ray, an excellent X-ray on the side there, that shows those dark areas, that black area, is an area that has actually been sealed off due to emphysema. So it's a non-functional part of the lung. There is air in there a little bit, but it's trapped. So it's a non-functional part of the lung. And here's what it looks like in real life. So the cavity areas, the large cavity areas, are areas that are sealed off. They're no longer functional parts of the lung. It's dead part of the lung. And you can see that that person's lost 30% of their lung capacity. A large percentage of it is no longer functional, which is quite significant. And I think I hit all three for that one, right? We're good? Yeah. Let me know if I miss anything. Okay, asthma. This one's pretty common. If you look at, I think do I have a blow up of it? No, this is blood vessel. Never mind. Thought I had a bronchiole here. I don't. I'm just going to jump to the next slide. I'll come back. But if you look at the cross-sectional area here, can I draw on this? Yeah. So if you look on the cross-section here, this pink tissue in the middle is actually muscle. So your bronchioles, the pathways that go to your alveoli, are muscled pathways. The exact function is unknown, why there is so much muscle tissue there. It may have been helpful in some earlier evolutionary iteration, but we don't really know entirely why there is so much muscle tissue in the bronchioles. It's not terribly helpful now for it to be there. In most people, it is not maladaptive, which means that it doesn't cause illness and you don't get constriction of those muscles for no reason. However, some people have much more sensitive bronchial tissue, and that muscle constriction can be activated by environmental factors or stress. And that's essentially what asthma is. So you're getting a constriction of the musculature around your bronchioles, and they are making the passageway smaller, which makes it more difficult to breathe. And when you do that, that straining and edema that causes also is exacerbated by mucus formation, which happens on the inside of the bronchioles, which makes it even more difficult to breathe. And you can get almost a full closure of the airways through an asthma attack, which is it's scary. It's scary. Question?
I heard that in like some of the puffers they have like a type of steroids.
Yeah, there's two types. There's two types. So one type is a steroid. That's a long-term treatment for asthma. And what the steroid treatment does is really reduce inflammation. It's causing decreased blood flow to the muscle. And it does have some tissue erosion where it actually causes a reduction in the tissue over time that is problematic. There are some side effects to long-term steroid use. It increases your risk of infection and a couple of other things, but it is effective at reducing the amount of immediate swelling that you get with an asthma attack. So that's a long-term treatment, but you don't use that in an emergency. I think that one is just used long-term. The other one that people have in the puffer, the one that you take in an emergency, is a muscle relaxant. It's actually the exact same chemical. It's like Robaxacet. If you've ever heard of that before, if you have like a back spasm or whatever, you can take a muscle relaxant. It's the same chemical, but it causes relaxing of the muscles in the airway, and then generally it opens them back up again. So those are the two main treatments. There are some other experimental treatments that are sort of in the process of approval. One of them, really interesting one that I heard about not that long ago, is the idea that they put like these, they're wires, I guess, down the bronchiole, and they heat up the outside of the wire, and it burns some of the muscle tissue. It's not a super high temperature, so it doesn't cause like scarring or anything on the inside, but the high heat actually like breaks down some of the muscle tissue and allows it to relax. I don't think that it's necessarily been approved for general use, but it's an experimental therapy, so that does exist. You can also take the immunosuppressant function is also caused by the steroid, so it also suppresses the function of the immune system. That's one of the reasons why it increases your incidence of infection, because it's an immunosuppressant. But you can't really cure it per se. For some reason, in the last 15 to 20 years, we've seen a significant increase in asthma, especially in young people in the population. We're not entirely sure of what's causing that process. Question?
Is it possible to have like a surgery to remove the muscle that's in the bronch?
It's part of the structure itself. So you have like hundreds of those pathways, right? And so you can't really take the muscle out of the middle of it. It's an interesting idea. That's really what the idea is behind the burning, which is you're trying to minimally damage the surface, but to break down the muscle layer that's underneath. I don't know how well it works. So certainly the jury's not out on that. But I've read a couple articles about the reason for the increase in asthma. And I think that the leading theory is the idea that we don't expose young children to allergens enough. In other words, we don't train their immune systems to accept allergens. We have a tendency in North America to hyper clean our interior environments. So kids have a tendency not to be exposed to environmental allergens, just things like soil and things like that. Kids don't go outside as much, and their interior surfaces of the house are often sterilized. And so their immune systems are just not trained to be comfortable with environmental allergens. And so when they go outside and are exposed to environmental allergens, they've sort of missed the window in youth where your body becomes acclimatized to environmental allergens, and then it increases the incidence of asthma as an older child and as an adult, which is interesting. There is definitely some evidence to show that that does happen. There may be other factors influencing it, though. Question? There are some genetic factors for asthma as well. So it's not purely environmental. There definitely are some genetic factors. Fair enough, and that absolutely exists too. So there are some genetic factors that predispose you to having more sensitive bronchioles, causing you to be more sensitive to environmental factors, for sure, or to stress. That question?
Does obesity have a relation with asthma?
It does. I think it exacerbates it. There are a number of reasons for that. One of them is the interplay. Fat cells are also hormone-producing cells. And some of the hormones that they produce can have a negative interaction, especially if you already have asthma. It can make your cells more predisposed to this inappropriate closing. And then the other issue with it is just simply the weight of tissue, which is that if you have a lot of weight of tissue sitting on top of your thoracic cavity, it just makes it more difficult to breathe. When people are obese, they often have trouble. You have seen this, people sleep with positive pressure machines. One of the reasons that you have to do that is because your chest is just so heavy that it's hard physically for you to lift it to breathe while you're sleeping. It's basically like the weight of yourself is pushing on it and making it hard for you to breathe. So there are a number of ways where it interacts, but I don't think it's not causal. I think it just exacerbates. Any other questions about these? It's interesting. Anyway, some triggers for asthma. There are a whole host of environmental factors, including stress, that can potentially be triggers. All right, let's talk about the itis, specifically bronchitis. Any itis, whenever you hear the word itis in medicine, means an inflammation of. Pancreatitis, whatever, any itis. It just means inflammation of that thing. Hepatitis, you name it. Fasciitis. So an itis is just an inflammation. A bronchitis is an inflammation of your bronchioles. Okay, so the tubes and the bronchi. And lots of things can cause inflammation. This is a pretty general, people kind of misuse this term. They say, oh, I've got bronchitis, as in I have some type of bronchial infection, but it doesn't necessarily mean infection. It just means that you have an inflammation. The inflammation could be caused by an infection, bacterial or viral, but it could be caused by a host of other factors. Exposure to heat, exposure to environmental factors, lots of things cause bronchitis. So it's just an infection or irritation of the tissue of your bronchioles. What does it look like? Cough. Usually a productive cough because there is mucus that's produced as part of the inflammation, and you have to cough it up. So productive coughs are often associated with bronchitis. Shortness of breath, wheezing. Wheezing is just having difficulty passing air through your bronchioles, and you get that when they're inflamed. Certain factors can exacerbate it. Smoking certainly makes bronchitis worse. Pollution can exacerbate it, and it can be caused by a whole host of things. Anything that's going to irritate your bronchial tissue basically can potentially cause bronchitis. And bronchitis, you'll note this diagram looks very similar to asthma. Yeah, the mechanism is similar. It's inflammation. Now, it's not muscle contraction in this case. You'll notice that the muscle layer is not actually like constricting here. This is just swelling beneath the epithelial layer of the bronchiole. So as opposed to muscle contraction, and there's usually mucus production as part of that. Any questions about that before we move on? Yeah.
What's the treatment?
Oh, treatment. Good question. You treat the cause. So if it's a bacterial or viral, well, viral you can't do anything. So wait. If it's a bacterial infection, you can give antibiotics. If it's caused by an environmental factor, stop exposing the person to that environmental factor. And that's pretty much the best you can do. So if it's caused by heat damage or pollutant that you're no longer exposed to, it's a wait and see. Hopefully you make a recovery. Yes?
If so many of these things are caused by like a buildup of like mucus and stuff,
Yes.
why don't they make something like you can take to like sort of slow like mucus production?
That is a great question. That exists. The treatments that we use regarding mucus in the respiratory system don't usually have to do with inhibiting mucus production. I'm not sure if that's physically possible to do. And it may, if you could inhibit it, it may cause problems because you'd be inhibiting mucus production everywhere. And so that might be problematic. But I don't know if it's actually physically possible to do that, or at least we don't know have a method. But there is a way to manage mucus, and that is by taking things that cause the mucus to be less sticky. You can actually get them as part of cough medicine. There's a name for it, for a medication that makes mucus less sticky, and it just makes it easier to cough it up. What is that called in cough medication? There's a name for this. Anyway, so we kind of do that, but that is a really interesting idea. What if you inhibited the mucus production in order to reduce the symptoms? There's got to be somebody looking into that. I don't know. I've never heard of a medication that specifically does that. There's probably a reason, but that's a really cool idea, though. Pneumonia. You guys have heard of this before. Pneumonia is an inflammatory condition of your alveoli. So as opposed to bronchitis, this is like alveolitis. Okay, it's inflammation of your alveoli. Usually when your alveoli become irritated or inflamed, fluid accumulates inside of them. Okay, so that's why the fluid buildup, the edema, is usually synonymous with pneumonia. When people talk about pneumonia, they talk about having essentially liquid in their lungs. That's because they usually come together. So what causes pneumonia? Again, just like bronchitis, anything that irritates your alveoli. So Bacteria is the most common, bacteria and viral infections. Fungal infections, not as common in humans. We do get them. There are some fungal infections that cause pneumonia, but not very many. Usually the fungal ones are for people that have a depressed immune system for some reason, because we're usually pretty good at managing fungus in the respiratory system. But you can also get it from chemical or physical injury. Again, inhaling something hot damages your alveoli. If you watch the video on the Tide Pods, that person gets pneumonia from, in that case, it's chemical damage to their alveoli, which again, can totally happen. But essentially, damaging the alveoli and then having fluid accumulating in them. What does that look like? You cough. It hurts. So it hurts to breathe in. The inflammation can be painful. Usually, if it's associated with bacterial or viral illness, there's a fever. And it makes it difficult for you to breathe. Again, it makes your alveoli less elastic, and so then the actual physical expansion and contraction of your lung tissue is more difficult to do. So it becomes painful to breathe. Can happen any age. When we talk about people dying of illness, young people and old people, and that is typically how pretty much every bacterial and viral illness that kills from a respiratory perspective, SARS, COVID is another good example of this. Those illnesses kill people via pneumonia. That's what causes the respiratory failure. So you can treat, usually you do treat with antibiotics, even if it's not bacterial. So normally you reserve antibiotics only for bacterial infections because it only kills bacteria. However, if you're having fluid accumulating in your lungs, you can get a bacterial infection even if that's not the cause of the fluid. So oftentimes they will give you antibiotics prophylactically as a preventative measure so that you don't get a bacterial infection, which can make it much worse. That's why you hear about people with COVID getting antibiotic treatment. They're getting it to prevent a secondary bacterial infection, not to kill the virus, which it doesn't do anything to kill the virus. There is also bacterial pneumonia, though, that is specifically caused by pneumococcal bacteria, which in that case, that is the cause. So then you would be treating the cause with antibiotics. But this is another wait and see thing. Pneumonia is so difficult to treat. It's not like you can vacuum out the alveoli. You know, this fluid accumulation, you just can't like suck it out of there. We're talking about like hundreds of thousands of tiny pathways. So it's more like could you suck the fluid out of a sponge? Not really. So it's quite difficult to treat. Here you're treating symptoms and then just hoping that the immune system is going to take care of whatever the causal factor is here. Not a ton you can do. There are some great, I should really look into this. More recently, we found a number of really good treatments for really bad cases of COVID. I'm sure you've heard of this before. So the actual mortality rate has gone down significantly. We've gotten better at treating it. Question?
For the project, could we do like say pneumonia and then like research about the treatment like of the antibiotic, or does it have to be an actual like machine, like an X-ray or something?
Interesting question. Could you use a medication as your technology?
Yeah.
I don't see why not.
Okay.
I think you'd be able to do all the different things. You could talk about the history of the development of it.
Because I'm just like thinking, I feel like there's going to be a lot of viruses or like infections that only treated by.
True, not viruses.
No, yeah, what's the word?
Bacterial infections.
Yeah.
Yeah, I don't see why not. I'm just thinking about the project about all the different parts. Yeah, I think that you could use a drug for all of them and it would work just fine. Yeah, so sure. In that case, you may want to focus in a little bit on specific antibiotics because they often have different mechanisms of action. So you might want to do that. Okay, what does it look like on an X-ray? There it is. You can see the fluid sitting in the lungs. Shows up pretty clearly there. Tuberculosis. We don't worry about this one in North America quite so much. Very common outside of North America. Outside of North America and Europe, but Central Asia, Eastern Asia, Russia, very common. Tuberculosis is surprisingly common. I don't know, I think maybe Sub-Saharan Africa is common for tuberculosis. I don't know what South America. Maybe. I don't know if it's endemic for tuberculosis. I'd have to check. But anyway, worldwide, this is quite a problem. So it is a specific bacterial illness. It's caused by Mycobacterium tuberculosis, a specific bacterium. And what it causes are something called tubercles. Tubercles are basically just clusters of bacteria. They're so big, the clusters, that you can actually see them. They form little globes of bacteria. Respiratory tuberculosis is the most common kind, and it's when the tubercles grow in the lungs. But you can also have nervous system tuberculosis, where the tubercles grow in the brain. That one's often fatal. And it is possible for one type of tuberculosis to turn into a different type. You can get the tubercles growing in your organs. You can get them pretty much growing all over the place. And the severity is different depending on the type of tuberculosis that you have. The respiratory type is the least dangerous and the most treatable. So because it's bacterial, we can mostly treat tuberculosis with antibiotics. There are some drug-resistant forms of tuberculosis, and they're becoming more common, which is unfortunate. We did talk previously about the problem with creating antibiotic-resistant bacteria. We're doing a terrible job with this, but they are becoming far more common. It's still in North America quite rare to run into antibiotic-resistant TB, but it does exist, so it is possible. We don't have a lot of cases here, so it's not a super public health concern, at least in Canada. Causes a cough. Oftentimes it's a blood-tinged cough because it damages the epithelial layer of the bronchioles and the alveoli, and so it actually causes like a little bit of blood to leak into the respiratory system, which you see in the sputum, the stuff that you cough up. It causes fever. It's a bacterial infection, so it activates the immune system. You get night sweats and weight loss. Weight loss is a very common symptom of tuberculosis. Did you guys ever talk about what's the play that you guys do in English class? Glass Menagerie? You guys do Glass Menagerie anymore? No, don't do that. That used to be common in high school. What was the name? What did they used to call tuberculosis? I gotta look this up real quick. You'll see it in literature all the time because it used to be extremely common in North America not that long ago. Of course, this is when my internet's going to stop working. Consumption. You guys may have heard that in a literary context before. No? Never? I haven't read enough mid-century literature yet. But if you ever do, often people will refer to somebody getting consumption. The reason it's called consumption is because people tend to lose a lot of weight when they get tuberculosis. But anyway, that what they're referring to there is TB. Here are some signs from the mid-century about not spitting. Don't spit in public. It can transfer TB to somebody else. It's spread by particulate from coughing and sneezing and stuff like that. It's not nearly as communicable as COVID, thankfully. So you really do need to like come into contact with sputum. You know, a little bit of aerosolized liquid from somebody's respiratory system. It's not that easy. You have to be in prolonged contact with somebody with TB to get it. So it's not something that you're just going to get from meeting somebody casually. But it used to be hugely problematic in North America. There were large public health campaigns around tuberculosis. It can be potentially fatal. Did I cover all three? Causes, symptoms, treatment? Okay. Question?
Yours says there's also asbestos on you.
Yeah, I get to that at the end. I think I circle back to asbestos. I'm fairly certain. Sorry if I might have them in the wrong order. Sorry. Oh, is lung cancer on there? No, it's not. Oh, that's why. That's what I did. So I actually mixed this up a little bit. Asbestosis causes lung cancer. So I'm going to discuss lung cancer here a little bit, and then I'm going to go back and talk about asbestosis specifically as a cause of lung cancer, because it is a cause of lung cancer. I realize this isn't on your sheet. You don't necessarily need to write this stuff down. Lung cancer, just like any cancer, is a malignant growth of cells. It is an uncontrolled cell growth, usually in a cluster, but this time it's found in the lungs. Obviously problematic. Tumors are bad everywhere, but one of the big problems with getting a tumor in the lung is that it's not simple to remove. So lung tissue, as you may already appreciate here, is full of blood vessels. That's the whole point of the lungs, right? You got to have a ton of blood vessels there for gas exchange. So cutting off parts of the lung are actually quite difficult. And so if you have tumors, you can't do too much cutting in there. You get prolific bleeding. It's very difficult to control bleeding in the lungs. So you can remove lobes. This guy. He doesn't have lobes. He's just got solid. Oh yeah, that's too bad. It kind of shows them on the back. I'll show you on the back. Can't really see it here. I wish I had a better model of the lungs. You'll be able to see it when we do the fetal pig. Yeah, we're on Friday. We'll do the fetal pig. I'll show you the different lobes. They're separate to each other. You can kind of see it on here. It just shows it with like a line on the plastic. But these lobes are actually separate. Like they have vessels that run in between them, and they're kind of separate. So you can kind of snip them off. So it is possible to get a lobectomy of the lungs and have part of the lung removed if you have a tumor in one location. Lung cancer tends to be very it's easy to metastasize. So it'll go to different locations quite easily. And so it's usually or often not isolated to a single lobe, so again, making it very difficult to remove. It also has a tendency to quickly metastasize to surrounding tissue, which again, hugely problematic. Once it leaves the lungs and goes to nearby tissues, like the brain, for example, it travels in the blood to the brain, extremely difficult to treat. So metastasis makes lung cancer extremely difficult to treat. It's also extremely painful. So lung cancer, obviously it's restricting your ability to breathe, which is tough. That's tough to deal with as a patient. It can be quite painful. It causes bloody sputum, lots of coughing and difficulty breathing, chest pain. You repeatedly get bronchitis and pneumonia, again, because it's causing inflammation in your alveoli and your bronchioles. And unfortunately, it is one of the most difficult types of cancer to treat. So if you look at deaths from cancer, I gotta look up the actual stats on this. I'm pretty sure that lung cancer makes up like 30% of cancer deaths. It's actually like a ludicrous amount. So far, far more than you would expect. A very, very high percentage of people that die from cancer are dying from lung cancer. And the main reason behind that is that it's caused by smoking. So it's one of the very few cases where people are intentionally exposing themselves to a carcinogen, because normally people avoid carcinogens. So it's like you know that this is a carcinogen, so it's like people aren't just like, you know, putting asbestos everywhere or whatever, at least not anymore, because people generally avoid carcinogens. But this is one unusual example where people are aware something is a carcinogen, but they still intentionally expose themselves to it. So it's obviously problematic. You'll note, by the way, that smoking only causes 50% of lung cancer, 50 to 60% of lung cancer cases, which seems weird. So, well, what are the rest? That stat, by the way, that 50% is a global stat. In Canada, I think the percentage is actually quite a bit higher than that. We have fewer environmental causes of lung cancer in Canada. There's not a lot of environmental pollution that's carcinogenic here. It does contribute a little bit, but there are definitely places in the world where a lot more lung cancer cases are caused by environmental factors. However, one of the factors that we do have a considerable amount of exposure to here is radon. This is something you'd never think of before. Radon is right here on the periodic table, number 86. It's a gas, it's a noble gas, and it's radioactive. And it's produced in some of the breakdown products when you get nuclear breakdown, fission of some of the larger elements at the bottom of the table there in the actinides. And those elements are found in granite and the Canadian Shield. The Canadian Shield has a lot of these elements in it, and their breakdown products are often radon when they break down. So one of the largest pathways to exposure, especially for people in North America in general, is from granite countertops. This is something that you'd never think about. But because those granite countertops contain some of those heavier elements, they actually leach radon gas. Not all of them. So some types of granite are more prone to this than others, but some of them leach radon gas. It's a heavy element. And so the gas has a tendency to accumulate in your basement. It kind of just sits there. It doesn't get ventilated very easily by your air conditioner or whatever, by your central ventilation system. So people end up having an accumulation of radon in their basement. You breathe it in, and then you're exposed to the gamma and the beta particles and stuff as the radon breaks down, as it radioactively decays inside of your lungs. And so that causes damage to your DNA in your lungs, and then subsequently can lead to lung cancer. So that is one of the other significant pathways in North America for exposure in terms of lung cancer. You can get radon test kits for your basement, just so you know. So this exists. You can just get them at the hardware store, but you can test to see if you actually have radon gas accumulation. It really depends on whether or not you have granite countertops and whether or not you live on the Canadian Shield. So there are parts of Canada where people actually live on bedrock. And in that case, if you're living on bedrock, again, you can get radon leaching that comes out of the surrounding bedrock and can leach into your house from the surrounding area. There is also a way to remove it from your basement. You can get radon abrogation systems, which pump the lowest level of air from your basement out of your house. So that also exists if you do actually have a radon problem. Just so you know, these are things that people never even think about. I certainly never thought about this, but it is one of the significant pathways for lung cancer is radon exposure in the home. Some viruses cause it, not a huge amount, but an asbestos particle. Do you guys know what asbestos is? Maybe I should start by just telling you what asbestos is. Why didn't that work? Uh-oh, did my internet get cut off? No. I don't want the size. There we go. That's a perfect picture right there. So what it basically looks like is like an arrow. Okay, it's like a spiky, barbed piece of mineral. It's rock. And that spiky mineral, because it has like all these spiky barbs on it, and these are really small, by the way. We're talking about like 3 to 10 microns, really, really, really small particles that are barbed. When you inhale these, they go into your alveoli or deep into your bronchioles, and they like wedge into the lung tissue. I just realized that this is on the screen anymore. Uh-oh. Is the thing still working? This is usually when everything stops working. Oh, it says excellent connection. Maybe it's just my iPad. I hope it's just my iPad. Has this been like this for a long time? Sorry, I didn't notice. It hasn't. Okay. All right. Nope. There we go. That's an asbestos particle. So again, it's a really barbed particle, very similar to like a burr. You know, if you ever like go in the woods and you get like burrs and stuff on your coat. It's like it's the equivalent of a burr, but like in a microscopic particle. And these particles just cannot be removed by your ciliated epithelium. And they have a tendency to burrow deeper and deeper into the tissue of the bronchioles and the alveoli. And they cause a ton of inflammation. They basically can never be removed. It's rock. So your body has no ability to sequester these or remove them. It doesn't dissolve into your blood, so there's no way for you to excrete them. And so they just basically permanently wedge themselves in between the cells of your respiratory system. And eventually, if you get that continuous inflammation over and over and over again, you get oxidative stress, all these other things that happen with continuous inflammation, and eventually it leads to abnormal cell growth. So you start getting DNA damage and other things from the constant oxidative stress from the inflammation. Anyway, so like I said, that used to be a huge contributor to lung cancer in North America, worldwide. But we virtually eliminated asbestos use. In fact, I don't think it's approved for any use any longer. But you'd be surprised how recently asbestos was used. When I started teaching, which is really not that long ago, 9 years ago or something, we had a ton of asbestos everywhere. In the chemistry lab, we used to use asbestos pads for heating things. We had an asbestos blanket that we used to use for fires. This is only 9 years ago, like not a long time ago. So we really, more recently, we've decided, oh, we should probably get rid of all this stuff that's full of asbestos. It's not really good to have it around all the time. And like I said, only 3 years ago, we had it removed from the ceiling. Now, I will say that the insulation asbestos that we used in the school is sequestered, right? Like it's not continuous with the air that we're breathing, so it really wasn't problematic unless you disturb it, like unless you go up into the insulation. But even then, you probably don't want that just like hiding in your building, just in case like some poor custodian is going up and repairing something in the roof, and then they're getting like showered in asbestos when they go up there. So anyway, we're very careful with this stuff now. It can cause quite a lot of problems. I think I have a picture of somebody with asbestosis. Give me one sec here. There's a person who's had a partial lobectomy. In other words, they've had one of their lungs removed. Usually they go in from the back, or at least an entire lobe of the lung removed. Usually they don't do the full lung. It's not that easy to get by with one, and for the purposes of lung cancer. So this just talks a little bit about the unusual accumulation of cells. The lung on the right-hand side here is a smoker's lung, and it has a number of masses growing inside. This is lung cancer right here. And some of these other parts, this right here is emphysema. This is emphysema here. So emphysema, and then we have some tumor accumulation. This person's not doing very well. I mean, obviously they're dead now, but they weren't doing very well at the time that this picture was taken. And it's really interesting when you look at the color of lung tissue, the difference. This is really like taken more or less immediately after death here. But this is a normal person's lung tissue. I mean, it looks completely different. It's really shocking to see how your lung tissue becomes discolored over time with continuous smoking. It really does. Like stuff accumulates in there. It's really crazy. But as you can see, like these people are, by the way, more or less the same age. I think it's like 40 and 42 or something like that. So they're very close in age. And so there's really no other factor here other than the smoking that's caused the difference to these lungs, which is shocking. And you can see how good the lungs are typically at removing particulate matter, assuming that you're not smoking. But like, yeah, they're really good. Like the lungs come out clean, even if you're like a fairly old person. Like, it's amazing how the mechanisms inside the lung actually remove particulate matter. They do a great job. We don't do this lab anymore. We actually used to go on a field trip where we looked at some of these at University of Guelph, and they don't run that field trip anymore, so we don't do that one anymore. But I probably should take that slide out of there. Just as a sidebar to all of this, cigarette smoking. These are there's been a huge change over the past 20 or 30 years. Thankfully, a lot less people smoke now than they did 30 years ago. When I was in high school, so when I was your age, which was 20 years ago, I worked in a restaurant, and it was so common for people to smoke. I wouldn't say 50% of people, but at least 30% of people were clearly smoking. And you used to be able to smoke in restaurants. It was gross. It was gross. You could taste it. And there's really no separation between the people that were smoking and the people that weren't. And so all the food had a little bit of cigarette taste to it. Honestly, it was super gross, guys. This is real. I cannot believe that this was in my lifetime. But people used to smoke all the time. It was super common. And a lot less people smoke. About 10 or 15% fewer people smoke in terms of the total population. So right now, it says 18%. I'm pretty sure that's more like 15 now. So it's gone down since 2009. Fewer people are smoking, thankfully. This is a much lower burden on the healthcare system as well, which is lovely because lung cancer is extremely difficult and expensive to treat, and we don't have to pay for that anymore. Yes?
I'm just curious, is it like the nicotine cigarettes that causes all this stuff? Or is
It isn't. Nicotine is not responsible for most of this. It's an interesting stimulant. People that take nicotine outside, removed from the context of smoking, don't experience any of these symptoms. I don't want to speak off the cuff here about all of the effects of nicotine because I don't know them all off by heart. But certainly, it's not responsible for the carcinogenity of smoking. The nicotine is playing other roles. It's highly addictive, but it's not causing cancer specifically. There's lots of other stuff in cigarette smoke that is. So people ask a great question, by the way. What's the difference between this and vaping? Right? People are like, oh, well, I switched to vaping. In general, if you're stopping smoking to start vaping, great. I'm glad you're not smoking anymore. It's certainly less hazardous than cigarette smoking is. That I can guarantee. Even preliminary data makes that very clear. It's much less dangerous than smoking is. But I think it's important for people to know that if you're not a smoker and you're just starting to vape for whatever reason, I don't know if it's as popular now as it was. It used to be so popular, not even just like a few years ago. I don't think it's quite as popular as it was. But anyway, if you're starting to vape, we really know very little about the long-term consequences of it, and it does cause inflammation in the lungs. So it's probably not free of negative health effects. What are the negative health effects? We don't have enough data to be able to say that very clearly. It's certainly less dangerous than smoking. That is a virtual guarantee based on all of the observations that we've already made about vaping. So if you're using it to transition out of smoking, good on you. I would say good for you. You should do that. But I'm not sure that I would start as a recreational activity. It's probably not. I would want to know far more about it than I currently do. And it's almost certainly not completely side effect free. And it is addictive, just so you know. If there's nicotine in there, oh, guys, I've never had to quit smoking before, but I know plenty of people that have. It is hard. It's really hard to quit smoking. Don't get started. Don't get started. It is tough to get off. All right. We talked about the cilia. Oh, there's an electron micrograph of the cilia, if you're wondering what they look like, those little hairs. So that's a photo of them right there. So you can actually see what they look like. So they just kind of grab little particles and they just pass them along up the respiratory system. I'm not going to talk about this. Skip it. You don't need to know any of that stuff anyway. Skip over the last part there. Any questions about any of these respiratory illnesses that we've discussed? Did I go back and talk about asbestosis? I think I did. I didn't? I missed it? So asbestosis is essentially respiratory inflammation caused by the inhalation of asbestos. So the cause is asbestos. You inhale it. It looks just like any bronchitis pneumonia. In other words, it has all the effects of bronchitis and pneumonia, where you are having a potentially productive cough, difficulty breathing. Sometimes you get blood in your sputum, although typically you don't. But it's like any general respiratory illness. But the end effect of it is that it often leads to cancer. Not always, but it has a very high propensity of leading to cancer. And the problem is there's absolutely nothing you can do to treat asbestosis. Once you get those particles lodged into your lung tissue, they're there. They cannot be removed. So, 10 microns, how are you going to remove that? That's no chance. No questions about this stuff? Okay, guys. So if you'd like, and I highly recommend this video because it's just fun, but you should consider watching the, where is it here? No. One more back. There we go. You should consider watching this Chubby Emu video. It's really interesting. If you're interested in medical case studies, this guy's channel is full of them. All kinds of crazy ones. What I like to do, my partner likes to do, I guess she's a physician, but we try and figure out what it is before they tell you. Often they won't tell you right at the beginning what's causing the symptoms. See if you can figure it out based on what you already know. Sometimes you can, sometimes you can't. But it's kind of fun. And then if you want, this is just purely for fun. If you want to see this asbestos video, this is like a commercial made by a company that produces asbestos, like showing potential clients all the wonderful things you can do with asbestos. It's from the 1950s. So if you're interested, at some point, consider adding some stuff to your vocabulary list today during Learning Block 3. But I don't have anything more for you. If you're still working on the project, the Meiosis Live project, try and get that in the next little bit if you can. I got to get your midterm reports out today. They're going to be pretty simple. So, are you attending? Are you handing things in on time-ish, relatively? And what's your current mark? You guys already know what it is, so you're not going to be like surprised by anything that you see there, unless you've handed something in recently. Okay. Let me know if you need anything. Try watching those videos; they're cool.